Okay, I'm going to begin here if everybody's ready to go. Okay, thanks. My name is Jim Walgenbach. I'm with North Carolina State University, an entomologist. And today I'm going to be serving as the moderator for the fifth and final stakeholder advisory panel meeting of the brown marmorated stink bug SCRI project. Due to the pandemic, this is the first year we've done this virtually. And the advisory committee early on kind of decided that maybe we ought to open up to a wider audience. So we did that, and I sent out an invitation to as wide a group as I could. And so essentially anybody who's interested in the brown marmorated stink bug from a research, extension, regulatory perspective, or pest management, and even growers, potentially are on this. And I think Kevin indicated we had about 165 registrants. So between that and the speakers, there's probably about 185 potential attendees here today. So that worked out pretty good. What I thought I'd do is just briefly go through a little bit of background information on the project before we get going. So I'm going to share my screen. Not yet. The pressure is always on when you're made the mistake of closing my presentation. So hopefully everybody can see these slides. So this project is supported by the USDA NIFA Specialty Crops Research Initiative. And to a lot of us involved in this project, it's kind of referred to as the BMSB2 project, because it follows the first project that was funded by BMSB, funded by the SCRI project from 2011 to 2016, which was led by Tracy Lesky. And the current one largely built off the success of that one, which was very successful. We currently have 45 co-PIs and 13 different collaborators. It's a large number of people involved, and that represents 18 different institutions and from 17 different states. Now, the funding period of this is such that it's a 5-year project that ends in August of this year. So that's why this is the last of our stakeholder advisory panel meetings. Now, traditionally, SCRI projects are not eligible for a no-cost extension, but due to the pandemic, that was kind of revised this year. So we do have a request in for a no-cost extension for this project. Now there's no additional funding that comes with it. So there's not going to be any additional research or extension activities beyond what we have planned through this year. Since most of the money is going towards supporting personnel, I think basically what this is going to mean, it's going to be a little bit more relaxed approach to closing out these grants. So,
hey, Jim, this is Larry. Can I cut in for a second?
Sure.
I was just going to let you know that I got notified yesterday by my grants office that the extension is complete.
Okay, great. So now we know. So a project this large, obviously we have to have a very good organizational structure, and that's been a real powerful aspect of this program. There, in addition to myself as the project director, there's 6 co-project directors that are represented by different regions of the state. You can see the different regions of the country, which you can see on the map listed here. In addition to that, each institution has a leader, and that person is primarily the contact point for budgeting situations, as well as providing annual report information. And then there's 6 project or objective leaders who lead kind of the planning of the various objectives during the course of the project. And then we also have an extension committee that consists of 7 co-PIs and 4 people from the Northeast IPM Center. And the Northeast IPM group are the ones putting on this webinar today and have been extremely valuable in all aspects of this. This project has 5 general objectives, including the landscape ecology, biological control, a management component, development of management strategies, and then an economic and outreach component. There's a lot of different sub-objectives under each of these, but these are the key areas that we're investigating with this current project. I also want to point out that we do have available to everybody an annual report from each year, and the one for this current year from 2020 that this meeting represents was just put up there last night by Kevin. And in addition to that, we also have from each of the stakeholder advisory panel meetings, presentations by each of the presenters during each of those meetings in past year. And if you want to find it, you go to stopbmsb.org, which is our kind of official outreach component, and you go to the About Us component, and you'll find out the reports and presentation section at that site. You can click there and you can see them from each of the 4 or 5 years. So today, hopefully you've seen your agenda items, and the meeting is going to follow where we have an accomplishment section followed by a stakeholder input section. The accomplishment sections will be divided into 3 modules, where largely based upon the objectives of the project. First, the landscape ecology and biological control objectives will be today, and then tomorrow we'll have the management component along with the economics outreach, and then followed lastly by the stakeholder input section. And we have the stakeholder input section is to get some information and feedback from stakeholders on the future direction of this project. We have some ideas about that, and so we're looking for input and discussion during that period of time. So during each of these accomplishment section modules, there'll be anywhere from 4 to 6 presentations, and I'm going to ask that people do not ask questions during that. Well, we're going to actually limit questions to written only. So you can ask them at any time in the written component, and we have 2 people who will be recording those questions. They'll be read, excuse me, at the end of each section during the discussion period when presenters can answer them. So again, just a little bit about the meeting protocol. We're going to ask that you submit the written questions using the Q&A option on the screen. If you're going to use the chat box, reserve that from maybe any technical issues you may have or private conversations. And then again, as I indicated, there's people that are going to be recording these questions, and they'll be read during the discussion period at the end of each module. And there will be opportunity for live in-person stakeholder input tomorrow during the input or the interactive session. So now throughout this meeting, following most presentations, there's going to be 1 or 2 polling questions that are posed to you at the end of the presentations by the speakers. And I really encourage everybody to answer those questions. If you don't think they're relevant to you, don't worry about it, because we're going to group the answers or responses into attendee categories. And we're going to put you in a category in a little bit based upon how you answer some questions here and some practice questions. So maybe we ought to do our practice polling questions right now, Kevin. And I've got 2 of these. First of all, what is your favorite specialty crop to eat? And this will be a good opportunity to see how long it takes everybody to respond. And I should point out that when these pop up, we're not going to read them. We're assuming everybody that's involved today can read on their own. So you can read it on your own and respond. How are we doing with our response?
Good. We have 81%. I think we can end it.
Sure. Let's see. And you're going to show us the answer. Oh, look at that. Apples and peaches. You must have a largely East Coast group involved here today. Okay, the next question is going to be important to answer, and I encourage everybody to do it. So if you could put that up, please. This is what we're going to use to categorize everybody's response into.
Actually, Jim, that question is in the next set after the first speaker.
After the first, okay, then we will wait on that. So well, that will be at the end of the regional session.
I believe it's at the end of the first presentation under landscape ecology.
Okay. we'll wait till then for that one. Okay, so just so you are aware, then when the polling question is going to come up, all the speakers' last slide is going to be the same, and it's going to be thank you. It's time for a few polling questions. So that will be your cue. The other thing I wanted to point out is that we're on a pretty tight schedule today, so try to answer your questions as quickly as possible. If you can do that within 15 or 20 seconds, that would be fantastic. Okay, at this point, we're going to go into the first session, which is traditionally the beginning of each of our stakeholder advisory panel sessions, where we get an update from the regional project directors on what this brown marmorated stink bug was doing during the past year in their region. And we'll start out in the southeast with Mike Hayes from University of Georgia. Oh, I got to stop sharing.
There we go. You all see my shared screen?
Yep, looks good.
All right, perfect. So Southeast, I want to highlight that we've got a number of scientists here in Georgia. We've got a big department of entomology. So props to Angel Eusebius Doria, Ash Cial, Brett Blau, and Schmott. Glenn Tillman is a wonderful collaborator that we've got here at the Tifton USDA ARS research unit. Rick Beslin's got some activity up at Kentucky, and of course, Jim is at NCSU. We didn't get nearly as much research and extension work done this year as we have in previous years, but I want to highlight just very quickly what we're doing. We did do a fair amount of trapping in areas that we had previously not to buttress our landscape objective. We did hold a number of virtual meetings to address those stakeholders, and this group did really well on journal publications. So I want to highlight that. In terms of actual research that we want to talk about, when we started this project 4 years ago, virtually every capture that we had was above what we call the fall line. So that would just be northern Georgia, and of course, North Carolina and Kentucky are pretty covered up. Here we are 4 years later, and we were asked to trap in areas where we probably would be the furthest extent of where we would see brown marmorated stink bug. And so you see all those counties that have a stink bug, those were all places where we did not expect them. In fact, when we started the project, when we had locations there, we had nothing. And we did capture bugs in all those locations. There's 2 locations actually where we got zeros, clear bottom center of the map, right on the Florida line, and then on the East Coast where there's a open circle there as well. Those were the only 2 places we didn't detect them out of all the places we searched. And we looked at a number of different crops, and these were late fall. So of course there were no immatures in there at all. You want to just kind of quick highlight the extension components of this. Given all these data that have been generated, each of the states have been really successful at extending that information to their growers. So Rick's group in the fruit and vegetable group, Jim's group with Cumberland Shenandoah, with the apple growers, with the peach growers, and then with the folks that we have in Georgia. We were fortunate that Angel was asked to do a keynote address for the Georgia pest controllers. That's 500-600 folks. So we had a lot of folks in one meeting there. And then of course the different commodity groups all got different sessions in terms of looking at the data and how that population is expanding. I had an opportunity to work with a number of really high achieving high school students that have been admitted to our flagship colleges for this year. And we spent a lot of time talking about invasive species. And I used our data sets to kind of educate them on the sorts of things that we do. And then the last thing I'll hit on here is we had a great year with publications, and I thought it really tracked nicely with project objectives that included season-long trapping as well as survival under different conditions, the chemigation versus foliar, so we get into some control, and then finally the biological control portion. So great year in the Southeast. Looking forward to the wrapping it up. And Jim, I'll turn it back to you.
Thank you, Mike. That was quick. The next one, let's go with Larry Goot, who's representing the Great Lakes or the Midwestern area. Larry?
Larry, I think you're on mute.
Unmute would be good. Okay,
There we go. Thank you.
slides are up. Thank you, David. So I'm responsible for giving the update in the Great Lakes region. We'll look at 3 states. First, Minnesota, and Bill Hutchinson sent me this information to update everybody on Minnesota with the initial detection in 2010. Now confirmed in 25 counties. You can see those in the map. A vast majority around the hotspot in the 7-county metro Minneapolis-St. Paul area still. Highest infestation in a place called Wyoming, just north of the Twin City metro. Detection is pretty limited in crops. However, Bill indicated they've now detected BMSB in 8 apple orchards. Okay, we'll take a look at Michigan. And this is data and slide courtesy of Juliana Wilson and the group that did all of the monitoring for BMSB across the state. We monitor in 4 different regions in the state, 58 sites using a sticky panel pyramid or rescue trap baited with a dual component pheromone lure with multiple traps at most locations. We caught 6,570 BMSB. And like every year that we've been doing this, counts are down pretty low until the beginning of August. And then not until the beginning of September do they start to jump up, and catching adults at that time. And you can see from that slide and from this slide that a couple important things. The first one, if you look at the different colors of these bars, you can see pretty clearly that most of the catch is in the southern part of the state. So the hotspots for BMSB remain in the southeast and the southwest. And here the green and blue bars are really tiny, almost non-existent. So once you start moving north in the state, catches are very low still. And this has been the case since we started this project, and it has not jumped very much during that period of time, although there's a few more reports in people's homes from up north. Numbers that we're catching remain really low. I wanted to show one more slide related to trapping this project. So we've been looking at all the different traps during this survey part of the project. We find a little bit different results than some of the other regions. We tried to make the panel trap a little bit better because the panel trap has been pretty much a bust for us. It's been not a very effective trap for catching, and it's not very consistent through the season. So we tried to make the post larger, and that didn't work. It actually made the trap not as effective. The LLIN insecticide netting trap didn't look as good as it has in the past. The last couple of years, I think this is more related to that it's hard to get it in an optimum position to catch because of the nature of the trap and how you have to deploy it. But the pyramid traps keep being the best trap for us. And we have excellent results for 5 years now with this rescue trap, this small version of the pyramid trap. The importance for the rescue trap, we believe, is that you need to have at least one of the fins touching some portion of wood, a post or the tree, trunk or limb, in order to allow these things to move up. If it's just hanging freely, catch is not as substantial, although they still catch. Mariana Zooks and her group at Michigan State, along with our group, have been doing a lot of work on releasing Trisulcus japonicus because we found it in the state a couple years ago. So in 2019 and 2020, we put out sentinel eggs and yellow sticky traps. In 2019, the sentinel eggs, we did not get any recapture of japonicus. Over these 2 years, we released 7,200 of them, adults. We did get some other parasitoids, you can see, but parasitism in these studies was always below 5%. In 2009, we did get one site with a positive attack of a egg mass that we put out with 7 individuals emerging from one egg mass at one location. Yellow sticky traps, we caught them in 2019 and 2020, just single individuals at 3 different sites. And then plans for next year. This just gives a recap of the three or four main areas where we've been doing these releases and where recapture has taken place, where we've released them. In the highest density site down in the southwest corner there, we have not recaptured any, but we plan to really work intensely in this area next year because this is really the BMSB stronghold in the state. Got slides for New York from Peter Jentz, and just going to show you, I think, one or two slides. He provided some slides of a survey, citizen science survey of BMSB in homes to kind of get an idea whether BMSB was going up or down in the state, and pretty much showed that most people have had pretty high numbers in their house, but if populations appear to be getting reduced, and this is clearer in this figure, where people in the survey, 61% said the increase that they saw before is now decreasing, and populations of BMSB are declining in homes across the state. They're also working on trisulcus, but I'm going to leave all that for Kim Homer because he has those slides and is going to update everybody on trisulcus for New York included. That's all I have, Jim.
Okay, thanks, Larry.
You told us to be quick, so.
Oh, that was great. Let's finish up on the Eastern United States and go with Tom Kuhar, who's representing the Mid-Atlantic region.
Okay, thank you, Jim. All right, I'm assuming everyone can see the title slide. So, yeah, I'm representing the Mid-Atlantic. I'm not gonna talk about research. We have a huge cluster of researchers that have been part of both SCRIs, and I'd be here forever if I was talking about all the research going on. So what I want to focus on, I think, is just regionally what's going on in the Mid-Atlantic. And I guess we're in a unique opportunity because we basically are the epicenter of, the first report of it in the US. And we've kind of had from that in the mid 1990s, they believe it showed up in Eastern Pennsylvania. But another big time period was the 2009, 2010, where just about everywhere in the Mid-Atlantic, in Virginia, West Virginia, Delaware, Maryland, Pennsylvania, New Jersey, we saw an explosion. In every one of these states, in most of the locations, we just saw the highest numbers of bugs that I think any of us have seen, and we've not seen that since. So I think that's important to note, that there was something that went on 2009, 2010. And what we're going to talk about now is just what we saw last year and kind of general trends. So I thought no better way to present this except for to just put some of the quotes. I surveyed my colleagues, and I'm not going to read them. You all can read the slides, but I will briefly summarize. So going from north to south in the Mid-Atlantic, New Jersey, it seems like they're seeing an overall reduction. There was definite comments made that things were pretty bad in that 2010 to 2012. Things like catching hundreds of bugs in light traps versus now they're only catching about 20 a night. 30% stink bug damage on peppers during that time, and now you're hard pressed to even find a damaged pepper. So these are kind of general trends in vegetables. Ann Nielsen is reporting that seems like populations in tree fruit are starting to increase in South Jersey, but that's tree fruit, and that veggies and field crops, we may not be seeing, many of these bugs, and that was probably much higher in 2009, 2010. Similar trends in Pennsylvania. They're just seeing lower reports of damage, lower crop damage. But Greg did mention that trap catch using the pheromone traps and the sticky panels was actually was high in 2020. So Ken Martin, and I really appreciate the data that he sent. He works with vegetables with Fermano Foods, and there was actually a survey of just a really good number of growers, Pennsylvania vegetable growers. So these are pretty good data over a nice span of 5 years or so. And this basically is all these growers responding to, whether they've had much stink bug damage on their vegetables. So tomatoes are a big one, but there's other crops as well, peppers, and you can see it there. And really the general trend is it's dropped. They're not seeing as much. Most of them are saying damage is less or the same, and very, very few have said that damage has worsened since about 2017. So these are kind of general trends. Again, these are vegetables, and this is Pennsylvania primarily. Maryland, similar case. Galen Dively, who's been working with this bug for the start, and he does talk to a lot of the growers, and he has some research plots that are going on with sweet corn and some other crops, and he thinks they've been low in recent years and probably 90% less abundant than they were in 2010 and 2012. So again, the same story. It seems like with regard to vegetables and a lot of these other crops that we've just seen a drop off. Delaware has definitely not seen anything like they got in 2010, and David Owens just thinks it's hard pressed to find any, and if so, it might be somewhere clustered around an urban area, or maybe an occasional tree fruit grower. So moving into West Virginia and kind of the mountains of the Appalachians in the western part. Well, Tracy Lesky reported high populations in 2020. This was based on a lot of trap catch and a lot of research plot work that they were doing. Also detected kind of a summer-long high of the bugs. Sometimes we see peaks, but sometimes we just see this. They stayed high the whole summer, and that was what she picked up in West Virginia. Similar results shown by Chris Berg in the kind of an area not too far from Tracy in the northern part of Virginia in Frederick County. And so we're seeing trap catch being high in those locations. I'm in the southwestern part of Virginia in the Appalachians, and we had very high trap catch as well. And I saw kind of definite some densities, moderate level densities attacking vegetables here. So there's variability, and that's kind of the point that I'm making, that in some areas they've just seen it drop and it's never been as high as it was. In some areas it looks like there might be some peaks again starting to show up. Chris wanted me to show some of these data, and I think they're good, just comparing 2019 to 2020. And these are sites where he's released TJ, the parasitoid, Trisulcus japonicus, and in those areas he's monitored brown marmorated stink bug activities. And you can see in several of the locations we've actually had more bugs caught in 2020 than in 2019. But some of the fields it was no change. So again, a little bit of variability. Now I want to mention, so these are all kind of mountainous areas or the northern part of the mid-Atlantic. When you get into the coastal part or the far eastern part in the same region, I think we've never had the establishment of the bugs that we've had in other parts of the state. And some of the results that we're getting, you can read these here. These are kind of more central or eastern part of Virginia, just low densities of bugs. I will mention on the eastern shore of Virginia that they caught the highest number of bugs in their panel traps than they've seen, but yet they're not showing up on the crops. So just a real mixed bag here, where if you're looking at trap catch, it looks like we might have had a peak last year. But if you're looking at other variables, they didn't seem to get into the houses or on the crops. So kind of some a little bit of confusing data, but I think a summary. So we're now looking at the end of 10 years, really, if you can't count both of these SCRIs. And what have we learned in this mid-Atlantic area? We had an explosion of these bugs in a lot of our states in the mid-Atlantic in 2009, 2010. It really brought attention. It brought these grants that ended up funding this project. But the one thing that we have learned is that we've not really had those densities since. We're wondering, are we going to get that again? What are the climate variables? Is it a dry summer? I will mention that one thing we saw is kind of the 2014, 2015, 2016, we did see a drop. And that did follow a polar vortex that came through. We had one in 2014, early January, and then we had it again 2015, same time period. And those extreme temperatures were lethal to the bug if it did not find adequate shelter. And so I think we do have severe cold effects that knock this bug back for a while. And then when we don't get that, and we may get some other climate effects, you might see these surges. I don't think we've got it all figured out, but we've been dealing with this bug for a while, and it's going up and down, but it's not been as high as we've seen in 2009, 2010. The other thing is that tree fruit seems to get hit a lot worse than these other crops. I think all of us would agree with that. We definitely have the parasitoid is popping up in more locations. That's really good news. Natural enemies are probably playing. The other big thing that we've learned is that organic farms and organic growers often have the worst problems with this. And I think it's related to the way their farms are often surrounded by trees and multiple crops. And I think it just creates a perfect habitat. And then you don't have the insecticide applications that are going down. So that's all I've got. Jim, I apologize if I was ran a little late there.
No, no problem. We're doing fine on time. We're going to go across country then. And maybe we can start out with Betsy Beers, who's covers the Pacific Northwest.
Okie doke, let me. Okay, which screen are you seeing?
We see your title slide. It looks good.
Okay, okay, good. Just want to make sure I had the right screen showing. Okay, well, so it was sort of a quiet year in Lake Wobegon. We, I think like most folks, we probably didn't get to do quite as much as we were hoping to this year. But most of us focused on, I think, key experiments and making sure that those got done. And I think at the same time, probably trapping and monitoring were a little bit lower in priority, and some of that didn't get done. So here you see the famous map of where BMSB is, and here's Washington and Oregon as an orange and a red state respectively. We're still in Washington, just at the ag pest and nuisance, well as Oregon a couple of years ago moved into severe ag pest. And that's based largely on their damage to hazelnuts. Here's a map of the finds in Washington just in 2020. And we've been keeping a database of BMSB finds in the state that go back to 2010. And the database is a website and an email address to which homeowners can report finds. And we integrate this with targeted sampling in my program. So in 2020, we saw a much reduced number of reports to our database, 61 reports compared to 146 in 2019. And realistically, I think, you know, people all over the state just had other things on their mind in 2020. Interestingly, 3 of the reports were from Chelan County, where Wenatchee is located, and they were at Vince Jones' house, my house, and my technician's house. So our chickens are coming home to roost. We had some additional evidence that numbers of BMSB were really down in 2020. However, here is 2 years trapping data in the same orchard blocks in the Mid-Columbia area. Note, while both were LLIN traps, they had slightly different designs, which also could have influenced capture. But we do think generally that populations were down in this area of Washington. So here's sort of our overall map showing the number of counties where there are currently detections. We're slowly filling in the state. Clearly, the heaviest concentration is on the west side of the state in the urban areas of King County, which has Seattle. And it's slowly but surely spreading in the eastern parts of the state as time goes on. So a general status of infestation in Washington and Oregon. This is, again, the Mid-Columbia area, which is encompasses Washington on one side of the river and Oregon on the other side of the river. In general, populations and damage have been growing over the past few years, but 2020 was maybe an exception in that it was a bit lower than some of the previous years. And again, the main crop here is, the main vulnerable crop here is pears. There's a new entomologist at Hood River, Chris Adams, who has started to keep track of brown marmorated stink bug, and he did some trapping in the fall of 2020. And they are definitely there in the Hood River Valley. They had one hotspot in a peach orchard, which is a pretty hefty trap catch for just a few weeks of trapping in the fall. And then in Southern Oregon, this again is a pear growing region. The general perception is that populations and damage have been building steadily over the years. But again, 2020 was just a little bit of a pause, a little bit of a low spot. And then the central part of Western Oregon, the Willamette Valley, this is where hazelnuts is the primary vulnerable crop. They do have some tree fruits there also, but hazelnuts is their big crop. And populations and damage have been growing over the past few years. This is where it's rated as a severe ag pest. And 2020 was, again, maybe not quite so much pressure as in previous years. So I suspect a lot of my colleagues are in the same boat I'm in. We didn't get as much done in 2020 as we would have liked, at least in the field. And I think both Washington and Oregon focused more on experimental work. And particularly, we focused on the release of T. japonicus. And again, Kim Homer is going to cover that part in his talk. So that was more the focus than either sampling for T. japonicus or trapping BMSB. And hopefully we will be able to return to near normal levels, research levels, that is, in the 2020 season.
Thank you, Betsy. Was that it?
Yeah, I just can't find how to unshare my screen. Oh. There we go.
Okay. Okay, finally, we're going to go to the last one, and that's the West Coast. And Diane Alston at Utah State University is going to give this presentation. There's a pretty extensive presentation about the situation in California that Kent Dano will present later. So Diane's going to focus on Utah. So Diane, your opportunity.
Thank you. Thank you, Jim. As Jim mentioned, I'm going to share the update from California and Utah. And also, as Jim mentioned, Kent is going to give a more detailed update on what's going on in California. So the slides I have here are very minimal, but just recognizing all the contributors to this information. So Kent shared some slides that show how the distribution of BMSB in California has changed since 2017. So just looking at the state here and the counties that show yellow where established and the gray-blue where it's been detected, you'll see how this changes over time with more counties being added. And he didn't share an updated map for 2020, so this is the most recent version that I have. As you've heard in past years at this meeting, almonds have been one of the primary crops of concern and also where they've seen quite a bit of damage. And so this is some information from Jalindra, Joanne, and Frank showing how BMSB feeding damage is definitely affecting almonds. Again, you'll get more specifics in Kent's update a little bit later. Also, pistachios, another nut crop that is of important value to the state, and BMSB has been a primary problem on. And also, Kent and Judith have been working with wine grapes as well. And then heading down south, Trisulcus japonicus was first detected in the state, in the southern part of the state, by actually Ricky, and I'm sorry I left Mark Hodel, should be on the slide, but he wasn't, and I forgot to add him. Also, Charlie Pickett's been very involved. And now in recent updates, they're finding detections of BMSB in pears in Northern California. And the California hotspots continue to be Los Angeles and Santa Clara counties and Northern San Joaquin Valley. So that's, again, a very brief overview of California. You'll learn more in a little bit. So a little update on Utah. So we've been certainly seeing some hot spots with damage, such as shown here in this peach from last summer. But as you'll see, I have some of the same trends to report that others have seen. This is showing some maps from 2017 and 2018 when we did plant host surveys. And in Utah, oriented north-south, it's called the Wasatch Front. So it's the western edge of the Rocky Mountains on the right sides of the map, and then the Great Basin heading out into the west or the left side of the map. And so the first initial detections were clustered in the Salt Lake City urban area, that corridor. And then you can see by 2018, there's a bigger spread, a bigger distribution of BMSB up and down that Wasatch Front, heading down to Provo in the south and up to Ogden in the north. But when you look at the size of the dots for the different valleys represented, you can see that already in 2018 numbers were less than they were in 2017. This is a map that's showing detections of BMSB from this past year. Doesn't show abundance, but a lot of these are findings in people's homes and in gardens, so not necessarily in the agricultural areas. So this is going along with some of the other reports you've heard from other regions of the country, where this is just total numbers of brown marmorated stink bug collected in a set number of traps between amongst these 4 years. And you can see this general decline from 2017 being our highest densities to much fewer in 2020. And the numbers of, this is for 4 different urban locations that have been our hot spots. You can see that our numbers are getting quite low. So this is some data from Lori Spears showing catches in agricultural places, so vegetables and tree fruits. Again, you've seen the same general trend with the hot pink line being 2017 and peaks in numbers in that year in July. They show weeks at the bottom, but that would have been in July, and then declining later in the summer. Whereas now when we get down to the yellow-green line that was this past year, the peaks don't really come until later in the summer, August, September, and then total numbers of abundance are much lower. Although 2020, we did see higher numbers in some of these sites than we did in 2019. So it's not cause and effect, but one of the factors that we certainly have been able to document is egg mass parasitism. So this graph is showing the percent parasitism of egg masses between 2017 to 2019. And the gray bars, which are pretty much non-existent, were the sentinel release lab-reared eggs that had very little parasitism overall, compared to the darker gray bars, which are the wild egg masses found, in situ on the different sampling hosts. And you can see a trend of increased overall egg mass parasitism over time. This breaks down these same data by species of wasps. And so in 2017 and 18, these different colors indicate these are species of native wasps that were found parasitizing BMSB egg masses. And then when we get to 2018, the red bar shows our first detection of Trisulcus japonicus. The black bar that's next to it are unknown because by the time the eggs were collected, the wasp had already emerged. It's a good chance that a strong proportion of these were also Trisulcus japonicus. So this is Cody Holthouse and Zach Shum, 2 of the graduate students that have worked on the project here in Utah in June 2019, with the confirmation from Elijah that indeed the wasps that they had found emerging from an egg mass were Trisulcus japonicus. The map on the right now shows for 2019 and 2020 the distribution of where TJ or Trisulcus japonicus has been picked up in Utah. So you see an expansion since that first detection in Salt Lake City area to up and down the Wasatch Front, where the brown marmorated stink bug is also being found. So just wrapping up, wanted to share some of the publications that our BMSB team here in Utah have put together. Up in the upper left, a host utilization survey that Cody Holthouse did as part of his PhD dissertation, parasitoid surveys in the upper right, and some parasitoid guarding behavior. That was some work done by both Cody and Zach. And then in the bottom right hand, a paper that Zach put out from his thesis that showed the impact on tart cherry, which is an important tree fruit crop here in Utah. It had not been really looked at in other studies or other states in terms of BMSB damage. And the bottom line is that it is very susceptible to fruit abscission between that petal fall to first blush stage. And so BMSB feeding early on would be the most disadvantageous to this crop. Generally, BMSB is not abundant in tree fruit orchards at that time of year. These were in cage studies where BMSB adults and nymphs were added to the cages at the different fruit development stages. But it does show a potential for high susceptibility and damage in those early stages. So that's just sharing some updates from Utah. And I will stop sharing.
That's great. Thank you, Diane. We're doing pretty good on time. If anybody has some questions for any of the speakers at this point in time, be sure to put them in the Q&A component of the on your screen. At this time, we're going to go to back to our map, our BMSB map. Share my screen here. So this is the most famous insect distribution map in the world, I think. I've seen this on so many presentations over the years. It's a good tool for us to get our message out. So what you see here, everybody hopefully is aware of this. This is shows you the distribution of BMSB throughout the United States, and it also incorporates into it a severity status of it. As of the last time this was updated, which was last October. Yep, I guess last October. There were only 3 states where it had not been detected, and I was made aware, I think it was probably about a month ago or so, that a detection was found in Montana, and it was actually made by a faculty member at the University of Minnesota, Amy Morey, who, interestingly, she went back to visit her childhood home, and that's where she found it. So that's kind of interesting. That was in January of 2021, and so that can go green for Montana at this point in time. So to my knowledge, South Dakota and Wyoming remain the only ones where there have been no official detections. They're kind of the outliers, as you see there. So I suspect they're there somewheres, but there's probably there's so few people, so much land, it's going to be difficult to detect them, I guess. So at this point, I guess if anybody has any comments about where, or if there's any updates that need to be made to this map in terms of the severity of the pest status, now is the time to speak. When will the map be updated? Here's a question. When will the map be updated to reflect Montana's detection? As soon as this meeting is over and Kevin can have an opportunity to fill in Montana.
Let's see, is Kim there?
Kim Holmer?
Yes, I'm here.
Sorry, Jim, I'm teaching a class on the side. My co-instructor is Kim, my bad.
Oh,
I
sorry.
thought this was muted.
Okay. So I know last year at this time, Nick Wyman is the one who had us make Oregon orange or red. So I guess there's no need for any updates unless I hear otherwise.
So there are a few questions coming in here now, Jim. Comments actually. Ontario should be switched to orange, but I'll check. EdMaps shows South Dakota as positive. And wondering, the last question from Don Weber, wondering if any sort of genetic monitoring of BMSB is ongoing because the source in China in particular range into the subtropics, and bugs that originated there might not be capable to colonize warmer areas. Certainly the global models indicate the risk.
Is there anybody who's been actively involved recently in genotyping populations from the United States?
Jim, this is Anne. We haven't done any work on it in the past few years. There was a recent paper a couple of years ago that did some additional analysis, included additional nuclear genes, but the populations do not look like they're coming from southern China, or at least based off the samples that have been collected so far. I don't think there's a lot of sampling in southern China.
Thanks, Anne.
This is Larry. I have a question for Diane. Hey, Diane, could you describe just briefly how you went in the field to find BMSB egg masses that were parasitoid in situ? And could you also send me the paper?
Yeah, Larry, I'd be happy to send you the paper. And it's really just a matter of looking. And for us, the northern catalpa is a real sentinel tree. And so that's particularly where we've had high success rates in finding egg masses and then finding parasitoids. And last summer, it was the first time, well, again, we only first detected TJ in 2019, but last summer, the graduate students were able to find wild egg masses and then also find Trisulcus japonicus, searching around egg masses and on leaves in the area. So able to actually see them in the field, not just, the parasitized eggs afterwards. So it was pretty exciting how well they seem to be doing, at least at this time, but happy to share the paper.
Thank you.
Okay, so I've got a couple of items here that people have pointed out that British Columbia should probably be yellow. Ontario should probably be orange. South Dakota is positive on the EDD maps. So Donald Lewis notes that Iowa should stay nuisance only for at least for another year. He's at Iowa State University. Okay, so there are a few updates here. That's great. Unless I hear otherwise, I don't see any additional comments. I think we can move on to the next component of this. I'll stop sharing mine. So at this point, we're going to move on to our first objective presentation, or first objective one presentation, and that's a landscape ecology, which is headed up by Dave Crowder of Washington State University. And Dave is, I don't know if Dave or Javier is going to be presenting, but the first presentation is the landscape modeling of BMSB, what we've done and where we're going, with Dave and Javi. So Dave, if you can share your screen and take over, I will let you know when you're 2 minutes up before your the end of your time. So you'll have 2 minutes to finish up when you hear from me next time.
Great. Okay. Can you all see this okay?
Yes.
Yeah.
Yes.
Okay. Let me know if you have any trouble. Today's a weird day. At 10 o'clock, I had to give a talk to the Pear Commission. I'm giving my class on this laptop, and I'm giving this talk on this laptop. So my class is in a discussion session for 30 minutes. So hopefully I have the right computers muted and not muted to do this talk. But I want to thank Jim for giving us the time and opportunity to update everyone on the landscape ecology section. This is objective 1 of the grant, and I think Javier is online here, and he prepared this PowerPoint that I'll be giving. Javier, as many of you know, has really been the leader in terms of the data synthesis of this big project. This is the largest landscape ecology project I think either of us has probably ever been involved with, and it's been a really nice collaboration. So this objective, I don't probably need to rehash it completely, involves more or less all of the institutions in the project, I think. This is the 4th year that we've had a really coordinated monitoring effort for BMSB, and we expanded it this year. And I'll show you some of the things that we're trying to work on now that we're at this phase where we have all these data. Sorry. So the strength of this part of the project from our perspective is really the collaborative nature of this team. I can't stress enough to the stakeholders that are here how well this team has worked together, how readily they've been willing to kind of send us data in a coordinated fashion. It's really worked quite effectively. And so we have this nationwide team that was about 17 or 18 different states we were monitoring for stink bugs from 2017 to 2019. And then this last year, we really extended the reach of our sampling to validate our models in some of the areas that are not a part of the project team. And so we were very fortunate, and Tracy Lesky has a big part in this, that Tracé, the company, were willing to give us free sampling materials to kind of expand our sampling to states that are not represented in the SCRI. So for example, we had monitoring for BMSB going on this year in Tennessee and Kentucky and Missouri and Arkansas and Iowa and Florida, and states that are not necessarily represented by PIs on the grant, but where we were able to find PIs to do extra sampling for us. And so the reason we did that this year was to really validate our models. We've been building these landscape ecology models, and I'll show them here in a second, based on the data where BMSB has established very well. And we've been trying to project ahead into areas where it might invade. And so we're kind of using this two-step process. So quickly, our sampling protocol has always been, we've kind of narrowed in on the sticky card traps of late. In the first year of this project, we were using both the pyramid and the sticky cards, but I think as a group, we decided that these were gonna work the best. And we've been placing these at the interface of natural habitats and host crops. And the strength of our sampling is that we have good temporal coverage of BMSB populations throughout the country that have been sampled from early spring into the fall in all of these different places. So I'll just walk you through. This is kind of a summary of where we've been over the last 3 years of this project. So in 2017, we had 18 states represented, 26 different research groups, 276 sites, and almost 25,000 BMSB. And you can see here the different sizes of the symbols represent where we were collecting the most individuals. And so we can see here, and I think I'll scroll through the other years, we see a lot of consistency from year to year, but I'll also point out some of the variability. So, you know, we always have high populations along the East Coast into Ohio and up into the mid-Atlantic. The populations seem to be bigger in 2018 in a lot of places. We didn't sample quite as many sites, but still over 260 across 17 states. And we collected about 5,000 more BMSB than the previous year. But we did see some shifts where you can see kind of, if you look at moving from 2017 to 18, populations got a little bit higher on the East Coast. But if you look at areas like the Pacific Northwest, 2018 was actually a lower year in Washington. And 2019 was a really low year in Washington. Things were picking up in Oregon. California kind of remained low, as I think Kent will talk about later. We had less BMSB in Ohio, but still kind of high populations up and down the coasts. And so this is kind of the summary of the 3-year data before this year. You can see this nationwide monitoring network. We have some kind of spots like Minnesota and Utah that are a little bit disconnected from the rest, but we have a lot of great continuous monitoring going on here that we've used to build into models. So what we did in 2020, these are some of the areas where we have data so far. If you haven't sent us your 2020 data yet, we're going to be sending out a call for the data. And we haven't got a lot of data yet from the cooperators in the other states, even though I didn't know they did collect BMSB. So our goal in 2020 or 2021 now, is to take all these data from 2020 and really validate the models. And I'll show it in some of the areas outside of the seaboards. All right, and so this is just showing, now I've overlaid, we have a paper that I think Javier is submitting this week that many of you are co-authors on. And this map here is showing from our previous work, where do we actually think where is it a good place to be a stink bug? And so you can see, we predict that it's a good place to be a stink bug, you know, especially throughout the Midwest into the South. You know, that habitat should get worse and worse as you move down into Florida. As I said, we have some sampling in Oklahoma and Arkansas. We're working with Danielle at Trace, you know, Tracy's former student. And she's collecting some insects for us in Oklahoma. We have some collections in Missouri from Kevin Rice's group. And so these are the areas where we're projecting BMSB to be reaching. We have some sampling in Iowa through Matt O'Neill's group at Iowa State. We have some work in Nebraska. And so, you know, as our models are predicting kind of, you can see this zone where we think BMSB could actually disperse from the from the east to the west. Now, it's probably unlikely that we're seeing BMSB moving through this range. You know, it's probably way more likely they're getting transported to new regions by humans, but you can see kind of a contiguous, you know, band of land that is a decent place to be a stink bug. I saw the mention about they were located in Montana this year. I think Billings is right around up here. Someone can correct me if I'm wrong. You know, we don't project a lot of Montana, the Dakotas, you know, past kind of the Twin Cities in Minnesota to be good habitat. It's too cold. So I am very interested in any collections we might be getting up in these regions, or if anyone's finding anything down in Texas and Florida or in northeastern US, that would be kind of outside of the range that we would expect. But to date, I think, and you know, maybe if Javier is here, he can say as well, we've been seeing that our models of where BMSB should be have been matching up pretty darn well with where they actually are being found. So some of the factors that we've been considering that may affect stink bugs at landscape level scales are climatic factors such as temperature in the summer and winter. So kind of hot and cold temperatures that might be limiting them at different parts of the year. Precipitation patterns, evapotranspiration is a measure of humidity. Growing degree days. I think Anne is going to talk more about phenology models, and we're really looking forward to working with Anne and her team and other folks in the BMSB project who have phenology models of stink bugs, because I think we can incorporate growing degree days more effectively into our modeling. And then some of the landscape factors, these have been adjusted over time based on conversations with folks, but include distance to water, urban areas, land cover, solar radiation, elevation, for example. So here's our maps. This is a paper that's going in right now. This is after all this work, we have some consensus models. Here's the situation as we see it right now. This map shows with increasing kind of redness or yellows, if the colors are getting hotter, it's a better place to be a stink bug. And so this is the map I showed before. This is how we think the climatic factors will change for stink bugs by 2050. So what might we expect over the next 30 years? And what we actually expect is, if you see here, a lot of areas in the southern US, the Southeast, these are the states like Mississippi, Alabama, Georgia, South Carolina, Arkansas, you can see have fairly low suitability right now. And as it warms up and gets dry and drier, we expect those regions actually to be a worse place to be a stink bug over time. So for those of you that are on the south, as it continues to warm up and get hotter and hotter and hotter, we might push stink bugs further and further north. As you can see over time, a lot of the habitat along the mid-Atlantic and up into the Northeast US is going to continue to stay very suitable. And we expect BMSB to keep pushing their way northeast. As you can see, some of these areas along the Great Lakes should be really, really good habitat for BMSB. And then we would expect them to kind of continue their march up this way. If you look at the situation in the West Coast, we're going to see similar things where kind of it's going to become more and more northward, the habitat's getting good. As you can see, we would expect more and more problems with BMSB up in northern Idaho into Montana, where they're currently not problematic. And so these are the shifts that we're expecting to see over time, that we're going to get hammered more and more in the Northwest. There could be some areas of California that get more suitable down south, but a lot of the Sacramento Valley is going to continue to be poor habitat for BMSB. And so we've been able to do a lot of cool things. And this is just showing the change in suitability here. I want to be clear, these are not exactly the same. So if you're in the Northeast or the Northwest, it's going to be a better place to be a stink bug over time. If you're in the Southeast or kind of the middle of the country, it's probably going to be a worse place for you. Some of the highlights of the work, and this is a paper we have submitted, we've really learned that the factors that drive the occurrence of stink bugs, and by occurrence, I mean, do stink bugs live in this place or not right now, are really different than the factors that drive abundance of stink bugs. So there is something fundamentally different about the environmental conditions that promote establishment of stink bugs. And that's a little bit different than the factors that once you have stink bugs that are established in your area, there are slightly different environmental factors that are actually fueling their populations. We do expect this to be a kind of evolving situation, both due to climate change, changes in landscape conditions, and then also just weather-related changes from year to year. The habitat suitability for stink bugs is going to change. Importantly, we have detected, I would say not even some, but many areas that we believe are highly suitable for BMSB where the species has not yet been detected. So as we continue to move forward, as we continue to expand this project, hopefully, we've identified a lot of areas where new monitoring should concentrate, and that's where we really concentrated in 2020. To show kind of overall where we're at, the main aims and project phases were really to predict the risk of invasion and abundance using modeling. This part has been completed. Exploring the most important environmental factors driving abundance and occurrence. This part has been completed. Now what we're doing this year is validating those previous models with the new data. We're validating both models of occurrence and abundance. And then this is what, if there is feedback, we'd like to really focus on this idea too, on what we want to do. Up to this point, we've worked on developing kind of a national map that shows risk of different areas for BMSB. And we've used all the data from the entire project to make those predictions. But now we really want to start tinkering with the question of, are stink bug populations acting differently in different regions of the country? And I know this has been an interest of the group for a long time. We've discussed how we might make those breakdowns. We decided biome was not very effective just because there were too many. But we need to figure out this idea of regional versus national models. And what I mean by that is, can we create one model that shows the suitability of habitat for the entire country, which is what we're showing here? Or do we need to build separate models because maybe the stink bugs in Jim's region are behaving differently than they are where Anne is located is different than where Betsy's located, and so on and so forth. So we're going to be going through this kind of iterative process now, and Javi is already working on this to try to tailor what is the best model for where you are. Do we need to base that just on sampling data from your specific location? Or are BMSB really kind of doing the same thing all across the country? And so this is going to allow us to determine if those populations are kind of behaving in fundamentally different ways. I've talked about in the past the modeling approaches we've used. We've used maximum entropy models to kind of measure where BMSB occur, and this technique called boosted regression tree. I'm not going to go into the details here. We're going to continue those approaches in 2021 as we evaluate, as I said, more regional models. So looking kind of across the different regions of the project, if it makes a difference, what type of approach we're using. I'm really excited to talk to Anne and others who are building phenology data. We've developed a new technique in our lab to directly incorporate phenology data into these kind of maximum entropy models. This is a new technique in the landscape ecology world that really seems to help you refine predictions of where species do well. And so as Anne talks about phenology and the data she's collecting and other people on the project are collecting, we're going to hopefully be able to use that data to refine our landscape models. And we still haven't done a whole lot with nymph data. A lot of what I've shown you has been based on adults, so we want to make better use of these nymph data. And we've started to do that in a couple of more specific targeted projects where we're working with particular groups, but we're trying to figure out what might be some of the best questions to ask with the nymph data. All right, so here's just an example of like a regional model. Let's say we want to predict BMSB in my region, the Pacific Northwest. We're going to be comparing approaches to say, what does this habitat suitability look like if we base it on all of the national data, or if we just base it on Betsy Beers and Nick Wyman's data, Oregon and Washington and Idaho data? Do we get a better prediction, or does it not really matter? And so, in theory, we will have the best models for what BMSB is doing in every single state, every single region of the country. And so, some of the conclusions of what we've done so far is that we have developed with a cooperation of many people, these niche models, they very, we think, accurately describe where BMSB occurs. You know, a model is a model, you know, no model is perfect. It's not gonna tell us everywhere that BMSB is. It's also not gonna tell us everywhere that they're not, but we feel like we have a very tailored, specific data-driven model that we can build off of, that we can both use to guide detections in new regions, but also kind of assess how we need to do sampling going forward and area-wide approaches as we move forward. And so we are worried, you know, it's not necessarily good news. We've detected a lot of regions that BMSB can move into. And so unfortunately, I don't think this is a problem that's going to go away on its own anytime soon. It's just going to, and I think, you know, Jim's and the map showed that it's just continuing to kind of slowly spread. So we need to stop that and nip that in the bud as much as we can. So some of the caveats in future research, I just want to mention a few things. Hopefully I'm on time, Jim. I think I'm about 20 minutes in, so maybe I'll have some time for some questions. But the results here are presented based on adults. We have data for all the larval instars for modeling. You know, perhaps this can be used to validate or test some of the phenology models. Perhaps there are fundamentally different questions about larvae. We worked with Jim's group a little bit looking at some of the data from North Carolina and just correlations between the adults and the nymphs. And it doesn't necessarily seem to match up perfectly in every case. So we really would like to know, for example, you know, if I have a set number of adults, does that really tell me how many nymphs I have? And it doesn't seem that it's a perfectly clear-cut relationship. I think part of that is that the traps we use are more effective at catching adults than nymphs. But part of it just might be phenological mismatch in some of the data. So I'm looking forward to using those nymph data more. And we haven't really looked at the temporal aspects of this. And I want to show a slide here in a second about what we might consider. But phenology models are not part of our current research. In my lab, obviously it's going on in the project. And we haven't really tried to showcase how the suitable habitat might change within a season. And I'm very interested to hear from stakeholders or other researchers if this would be useful, if we could maybe show, you know, here's what the suitable habitat in, say, North Carolina looks like in May, and, you know, how's that going to change in June and July and August and so on. We could have more of a temporal dynamic to this to show how landscapes might change in ways that make them more or less suitable to stink bugs. Future work that our lab is doing in collaboration again, and I'll just show one graph of this. We're very interested in taking the data that the project has collected. We've worked with Anne's group at Cornell to gather data for Nosema, and I will show that data on the maps in a second. We're almost done building niche models for Nosema, and that was the goal with the work with Anne Hayek's group, was to collect populations of Nosema all throughout the country, and then use these same approaches to find out where is it a good place to be a fungus, and then where is it a good place to be a parasitoid. And so we have in our possession all of the data collected from the project on TJ and Nosema, as well as all of the data that's ever been collected on TJ outside of the project. Where does it like to live? This is just showing you, for example, where we've gotten data for Nosema. And you can see in the red, it tracks pretty well kind of a lot of the areas where we've been sampling for BMSB. So hopefully, you know, Anne's lab has tested in all these areas, you know, how infected the stink bugs got with Nosema, you know, and so we're going to really be able to look at, are there areas where biological control seems to be working really well? And are there areas where it doesn't seem to be working so well? Similarly, we're going to be able to put Trasilicus on the map and show how they're doing. The approach that we're going to use, and I'm going to go backwards just a bit, and I hope this, is to show maps like this, where you could imagine if we take a map that shows BMSB and we merge it together with a map that shows Trasilicus, it will show something like this, that, where is the overlap highest or lowest in kind of where biological control might be effective. So this is somewhere we're going to move forward in the future. Finally, I have a lot of interest, and my lab has been working on a project to build up spatial tools for pests. And this is something that I was thinking we might do with BMSB if it's interesting to folks. In my lab within Washington State, we have a monitoring network for insect pests where, just like within the BMSB network, people are out there collecting data, and they're collecting it every week at a bunch of different sites. And we've created a website, and I'll talk a little bit more about this tomorrow in the stakeholder group, but I want to mention how this works. Members of our lab or extension scientists go into the site and they enter the data. In this case, we're looking at a potato psyllid. And then we use these kind of landscape tools to show at any given time, where are the insects occurring? Where are they kind of at moderate, high, very high levels? And it's not shown here because this is just a screenshot, but if you are on our Potato Decision Aid website, there are new maps of this insect created every single week based on our sampling data. And the growers and consultants can kind of look and see as the insects are moving around in the landscape, as they're coming out of overwintering sites, as they're moving into crop fields. And this serves as an early warning tool to kind of gauge when those populations might be arriving. And so I would be very interested in thinking about how we could build over the next year and a half cooler tools for growers to use, working with the extension team or whoever else to think about what could we deliver to our stakeholders that would actually help them make some more proactive decisions regarding BMSB that are informed by our landscape ecology studies. I think we've done a great job on the basic ecology. And now we need to really work on the delivery. How do we translate all of this complex information we have into something simple like this, where in theory, a grower could, these red dots and green dots on this map show weather stations that I've set up just because I'm interested in those locations. And if I were to click on any of those points on the map, it would take me to a little pop-up that says, psyllids are at moderate levels in your region. Here's the thing we recommend you do to manage psyllids. And so I could see a very similar system being developed for BMSB based on the type of monitoring we're doing that could really hopefully guide area-wide pest management. So I just wanted to close with this just to show you, landscape ecology can be very complex. There's a lot going on, sometimes kind of in a black box that's hard to understand, but our ultimate goal is to turn it into something simple that growers can actually use to make a better decision. And so that's where we're at. And I think that's all I have, Jim. Hopefully I was on time, and I don't know if you're taking questions or if I'll just stop there.
No, you're doing great on time. Dave, if you can stick around till come either that or come back at the end of this session.
I'm going to be here all day, Jim. Yep.
Okay, so we'll hold the questions until the end of each section.
Great, sounds good.
Okay, we're a couple minutes ahead, but that's fine. I suspect we're gonna need this little bit extra time later on in the program. So,
Jim, we want to do a quick poll.
Oh, okay. Do you want to do that now? Sure. Okay. Yep. So everybody can read this on their own. I see no need for me to read it to everybody.
I would encourage the panelists to vote as well here. You should have access to the polling.
It might not be clear, but the difference in 1.2 between number 3 and number 4 is number 3, you are officially affiliated with this SCRI project, whereas number 4, you're not. So.
Jim, I just want to add one thing while we're doing this poll. My lab will send out in the next couple days a list of all of the data we have so far from 2020, as well as all of the data that we're using for TJ and NOSEMA. And if we'll send that to everybody, and if anyone notices that we're missing their data, please contact us so we can get it in our databases. And we'll be reaching out to people to get their data. But if you think you've sent it to us and it's not being represented, we definitely need to know, because that means we missed your email.
Okay. So have most people answered these questions, especially number one? That's pretty important as we move forward.
We're up to about 65%.
Okay, I know that I just tried to answer, and mine, it does not have the option to submit for some reason. I hit submit and it doesn't do anything.
Oh.
Did you answer all of them? Sometimes you have to answer all of them before you
Oh,
can
I didn't
submit.
see that there was a last one there.
Yeah, so as everyone scroll down, there are a few questions, and then there should be a submit button.
Sounds good.
Okay, thank you, everyone.
Thanks. Okay, we're going to move on now. The next presentation is by Ann Nielsen, who's going to talk about advances in modeling of BMSB phenology. Ann, you're on. Just as a reminder, Ann, I will chip in after when you have 2 minutes left.
Okie doke. Is it sharing my screen properly?
Yes.
Yeah.
Okay.
Yep.
Is it showing the preview slide? Because sometimes it switches back and forth.
It looks good.
It's
Okay.
showing your title slide, Ann.
Okay, thank you. So I'm going to attempt to summarize multiple years of work by multiple labs, and including some ongoing research right now. So this is work being done by myself. Then my postdoc, Robert McDougall, has been contributing and writing up and analyzing some data on pre-oviposition, and specifically how it relates to diapause termination cues. Shelby Fleischer at Penn State and Shi Chen at UNC Charlotte are part of our modeling team. And but when I say part, it's the three of us, and Chen is our modeler. And Jim Walgenbach and his lab have also been doing some work on the diapause termination cues, as well as validation of some of these models. So, and the hope is that we will kind of merge all this together with what David just talked about and create some really, really dynamic and accurate predictive models. So when we talk about a model, when we're talking about a degree day model, we usually refer to these as these kind of linear approaches, typically for our moth pests, right? So we have an infield biofix, and then we have an oviposition event, and everything's done based off degree day timing and trap catches. And we have these discrete and distinct peaks in our population abundances that we can really distinguish. This is first generation, this is second generation, third generation, and so on. And we can use our pheromone traps or other phenological, plant phenological events to time our management. And these work really, really well. They're simple to use. They interface really well with weather-based software. But they do fall apart when you have overlapping life cycles and overlapping adult generations. And that's exactly what we have with brown marmorated stink bug. We also aren't using a sex pheromone trap, we're using an aggregation pheromone trap. So these factors all make a standard kind of more linear degree day model less accurate. And it doesn't allow us to incorporate other factors such as mortality, landscape variation, like Dave was talking about earlier, or any physiological variation between populations. So what we did, what Shi Chen, Shelby Fleischer, and I started doing a few years ago, is to develop an individual-based stochastic stage-specific phenology model. So that's a lot of words there, but basically it allows us to have variability between survivorship, both at the adult stage in terms of overwintering survivorship. It allows us to have a variation in our survivorship between eggs and nymphs at different temperatures and at different life stages. And it allows us to incorporate our standard degree day developmental requirements. And it also allows these variations between the populations because it is done for each individual. We're allowed to put different parameters on the kind of life cycle of this insect. And we can also put into the standard models in terms of fecundity, the time to oviposition, et cetera. And one thing that's really important for brown marmorated stink bug is that we put in diapause termination and induction cues. Because this is a bug that's overwintering in the adult stage, we have determined that photoperiod is the primary cue for most of our populations, at least, to terminate diapause. So it allows us to put all of these different components into the system and not only predict just when the population peaks or when the phenology peaks are going to be for each generation, but also which generations overwinter and what kind of we see in terms of the population growth over time for each of these locations, each of these individuals. So we ran this model with 1,000 different individuals, and we pull these results from 100 different simulations of the model run. And what it kind of generally looks like here is that we start off with a non-reproductive female. This is a female that's overwintering in diapause. She becomes reproductively mature as a function of degree of photoperiod. So we know now through laboratory work that 13.5 hours of photoperiod is about when 50% of the population becomes reproductive. So that's a really good biofix that we've been using. You have heard us probably talk about 12.7 hours of photoperiod as a biofix as well, and I'll kind of circle back around to that as well. So they need an interaction between both photoperiod to start reproduction and then additional degree days in order to become reproductively mature. Then those females become reproductively mature, and then the eggs and the nymphs develop and become previtellogenic or not as a function of degree days. So we know the degree days based off of our traditional developmental rate models. But then at the end of, once a female becomes an adult, she has the kind of decision based off of climate factors to either go back into this cycle to continue a next generation, become reproductive and produce offspring, or she goes into diapause and is a non-reproductive female. So we have these kind of two options here when we have a diapausing female. We also can incorporate overwintering survivorship, and that's data taken from Tim Hayes' paper in Switzerland on mortality or survivorship of those overwintering bugs, and we can take into developmental survivorship. Another factor that we don't take into consideration here is whether or not the photoperiod is different between locations. We don't take into account if the time to become reproductively mature is different for first generation adults or second generation adults. So there's a lot of variables that we can still tweak in this model, and there are relatively simple ways to change that. So we ran this model at 8 different geographic locations throughout the US. These were all selected because they are key agricultural areas. So we have, Wenatchee, Washington for our apples. We have Salem, Oregon. I think that's because of our wine grapes. Davis, California, Riverside, Homestead, Florida, Asheville, North Carolina, Bridgeton, New Jersey for our delicious peaches, Geneva, New York for our apple production again. So these were kind of all coastal areas, not coastal, but East Coast, West Coast. At the time, that was where we were concerned. So we could add additional locations like Michigan and Utah, for instance, to this model and run it again. So we get a lot of different outputs from this model. I'm just going to show you some outputs on the adults based off of the total number of adults per generation. So what we get is, we ran this for 10 years. So we were running this model back in 2016. So we did the data from 2005 to 2015 at all of these locations. We start off each year with 1,000 adults that are coming out of overwintering. That's our parental generation, or P in black. Then if those produce offspring, those become the F1 adults. And then the blue is the F2 adults. So this indicates that in Bridgeton, New Jersey, which is where I'm located, and we produce the great peaches of New Jersey, we have 2 generations per year. You can also see that we get really big population peaks here in 2010 and 2011, and those happen to coincide with years that we had big populations in this area. So we kind of know anecdotally that the model has validity there as well. One thing I want to point out, if you can see this, is that there's a lot of overlap here between that first generation and that second generation. And that's what I was referring to earlier when I said that our traditional degree day models aren't as effective at distinguishing between first generation and second generation adults. And in the field, it's really hard. You can't just pick up a bug and say, oh, this is a first generation female. So we do get this overlap, and a lot of those big peaks that we see, what we realize are actually second generation adults. So if we look at Asheville, North Carolina, where we actually have similar degree day accumulations, we see similar trends. But we can see that there is some slight differences. We have differences of about 3 weeks or 2 weeks, I think it is, for the start of the biofix. So there's a little bit difference in terms of the time at which BMSB is active in the field. And we get slightly different mixes in terms of the overwintering, who overwinters at the end of each year. Now, if we go to a population with a similar latitude, which Davis, California is actually at the same pretty similar latitude as Bridgeton, New Jersey, which means that they have the same approximate date of that 13.5 hours of photo period. You can see here that, again, we have 2 generations per year. That F2 generation is really big in most years. But one thing I want to point out here is that the population size in Davis is very different than the population size in Bridgeton. Now, this does not incorporate other factors such as humidity effects. It doesn't incorporate factors such as possibly differences in diapause state either, or landscape variables. So this is based on kind of what David was referring to as that national model. So we took the same developmental parameters for the entire population and applied them based off of the individual degree day or weather accumulations at each site. When we look at this for all of the different sites that we modeled this for, you can see the maximum adult population at each site. So you see a very high population in Homestead, Florida, which we're not really seeing. And we're seeing a really large population in Davis, California, which we've seen in some years and not in other years. The populations in Geneva, Bridgeton, Asheville, Wenatchee, Salem are all kind of at this moderate level. So we do see the model does predict really big differences in stage structure between locations. And by that, I mean the size of the F1 versus the F2 and which of those generations becomes reproductive and overwinters. It also, we can see here that we see very large differences in the population size between all these geographic locations, as we would expect. And it does suggest that partially because of this difference, but also because of the mismatch that we maybe see as predicted from the model and based off of what we're seeing on the ground, that landscape features or other factors may be playing a really important role in moderating these populations. So we have validated this model in a couple different ways. One was a really robust analysis by Angel Aceves-Doria, who took pheromone trap data for 2 different types of pheromone traps, our sticky panel trap and our pyramid trap at multiple locations. And we compared them at select locations, Michigan, Maryland, Georgia, and Oregon, between what the phenology model predicted and what our peak trap capture was. So if you look at this table here, you can see that for adults, we don't see any significant difference between locations, which means that the model, even though sometimes it's off by about 16 days or so in terms of the peak, it's not significantly different. We do start to see some significant differences for our predictions for the nymphal populations, especially in Michigan and Maryland, as well as Oregon. For the pyramid trap, there's not any significant differences with the clear sticky trap. So that there could be a lot of factors that influence that, but it also tells us we need some further refinement of our model or placement of traps or how we're sampling. So Jim Wagenbach ran the model with some field caged adults as well. And he has overwintering adults here, or the parental generation in red. Then they tracked the eggs of those adults and then each generation. And he did this at 2 different locations in North Carolina, one in Mills River, which is at a higher elevation, and then Goldsboro, which is at a lower elevation, more coastal. And we do get a second generation of adults at the Goldsboro location, but the Mills River location doesn't seem to be producing eggs in his field study. But this is using that 12.7 hours of photo period as a biofix. And that's from a different paper where we dissected field caught females throughout the country and assigned them to different reproductive rates. And 12.7 hours was the point at which all of those populations best convened on. So if we model each of those differently, we get very different rates. And so that's just based off of activity of the adults in the field. But 12.7 is the point at which I think it's 10% of the adults are reproductive, whereas 13.5 hours is 50% of the population. So I would still lean towards 13.5 hours of photoperiod as a biofix if we're talking about 50% of the population. So when Jim ran this in terms of the cumulative degree days from April 4th, which is the time point at which he gets 12.7 hours of day length, you can see that there's a really good fit for oviposition and a really good fit for F1 adult eclosion. So we're having R-squared values of 90% in both of those locations. So the model is fitting his field data very well using that biofix of 12.7 hours. So going back to our phenology model, we have a model that works, that's predictive in a lot of locations. It is consistent with our trap captures at multiple locations, at least for the adult stage. There's of course always ways we can improve it. But one thing we started working on together was whether or not we can add in biotic mortality, specifically parasitism and predation. So now we have our Trissolcus as a biotic mortality factor as well. And looking at the first run of results, we have parasitism at 0%, 20%, and 40%, and this is data pulled from the literature. We also did predation at 10, 20, and 40% as well, or at least 10 and 20%, and it didn't impact the population as much as we would expect. So here's what the populations look like for Bridgeton and Asheville at 0% predation and 0% parasitism. If we have 20% parasitism and 40% parasitism, you can see significant impacts to those populations. And that's just assuming that 2-day window or 2 or 3 day window for parasitism by Trissolcus japonicus. So if we can get up to 40% parasitism, I think we'll be in really good shape for impacting BMSB. Of course, this model still needs some tweaks, but I think it's a really good next step to figuring out some of these our phenology models. So our next steps here are to incorporate any geographic differences in phenology, including overwintering survivorship and critical diapause cues. We need to refine the impact of biotic factors and landscape influences. And then working with Dave's group, hopefully we can kind of combine the landscape Climax Maxent data that they have with the phenology model so that we can develop really a landscape, very site-specific decision support tool, and maybe even incorporate things like parasitism or management into it. So that is all I've got.
You have your next slide?
No? Was I supposed to have a next slide? That's it.
Why don't you throw up the questions, please?
Is there a question from Coldy Holthouse?
We will answer questions at the end of this.
At the end. Okay, thanks.
Okay, let's move on. Our next presentation is going to be by Betsy Beers, who's going to talk about the penalty box, the high cost of growing up in Eastern Washington's native plants.
Okay, is the right screen up?
Yes.
Okay, great. So yeah, I'm going to talk a little bit about basically dietary quality and its effect on brown marmorated stink bug. This, first of all, this is just sort of the satellite view of Eastern Washington. And what we're talking about here is the shrub steppe habitat. This is this region both in Eastern Washington and in Eastern Oregon. It's the rain shadow of the Cascades. And as you can see in the satellite map, it's that big brown area. You also notice that our BMSB database shows most of the finds in the wetter western parts of the state, with just pockets in the eastern part, and most of those pockets really just reflect our sampling effort as opposed to the natural establishment of of BMSB. But we wondered at the outset of this project if our arid climate is intrinsically less suitable than other better rain-fed climates in the state or elsewhere. We also noted that, in fact, we have lots of native stink bugs in Washington, a handful of which occur in that dry zone and are known pests of tree fruits. So from previous work, we know quite a bit about which of our native plants are used by our native bugs. And it seemed like the most logical place to start. If it's a good host for a native stink bug, maybe it's a good host for brown marmorated stink bug. Previous work in the East clearly points out the importance of fruits or reproductive parts of the plants as the most likely to be fed upon. So this is these are the plants that became our target assemblage for some dietary studies. When you look at a hillside or a plateau in eastern Washington, the vegetation can look very, very monotonous: sagebrush, bunchgrass, and sometimes that in fact is pretty much it. But at higher elevations, in ravines or riparian zones, anywhere there's a little extra water, we have a surprisingly rich array of native plants that grace our landscape. So we did a series of growth room studies in bug dorms to look at nymphal development and see if it was affected by diet. The first study compared the native plant assemblage to our colony diet, which is a typical potted green bean, carrots, and nut and seed mixture that I think many of the lab groups use. So part one of this study was done in the early part of the season, and part two was more later summer into the fall. And it's trying to mimic when the two generations of BMSB might be present in our area. We speculated that plant quality would be different during these two periods, and senescence in the fall, in fact, might impinge on those overwintering adults. So we followed the development of nymphs from egg to adult, noting developmental time, survivorship, adult weight, and sex ratio in these two diet groups. So here are the results for part one. And this early season study gave us a very clear answer to our hypothesis. The nymphs took slightly longer, but not significantly longer to develop, and produced significantly smaller adults on the Western diet than on the colony diet. The biggest penalty, however, was in survivorship. And this was reduced nearly 43% in the Western group. Part two, the late season study, gave us essentially the same answer. In this case, the developmental time of both males and females was significantly longer in the native diet group. But again, the biggest hit was in survivorship of those nymphs. Not that many made it through to adulthood. So encouraged by the very clear results of these 2019 studies, we used the same basic format to refine this question. The first element of this was changing our comparison from a colony diet, which BMSB does not necessarily encounter in nature, to a plant assemblage that was more typical of the Mid-Atlantic, where we know they're well established and appear to thrive. So I asked advice from Eastern colleagues as to the composition of this plant assemblage, but in the end, we did have to use those plants that were available locally in the form of residential landscape plants, or the ubiquitous and invasive tree of heaven. And although the variety of plants in the Western diet looks greater, these were sort of sequenced through the study depending on plant quality or time of fruiting. And usually there were never more than 3 available to the nymphs at any one point in time. So my assumption was that much of the difference between the two diet groups would diminish or even disappear when we substituted the Eastern diet for the presumably ideal diet that we feed our BMSB in our colony. However, this was not the case. The differences were still stark. The Eastern diet group fared far better than the Western group, which had longer development developmental times, lower adult weight, and as previously, there was a huge survivorship penalty. This is about 50% lower in the Western group. So yes, BMSB can develop on our native plants, but they certainly do not thrive on them. Now, up until this point, we had focused on nymphal development and used adult weight as a proxy for adult reproductive performance. However, it seemed prudent to test this. So we had an opportunity in 2020 to continue these studies on. We took 10 pairs of adults from our Eastern and Western diet groups, put them in small cages, and followed their fecundity and longevity through their lifespan, but keeping them in the same diet that they had grown up on. The adults we picked at random that did not in fact have significantly different weights going into the study. And yet the trend for poor performance on the Western plants continued through adulthood and in fact throughout their lifetime. The fecundity of the Western females was greatly reduced and the lifespan of the females, surprisingly, but not the males, was also much lower. As some of you may remember from last year, I'm a big fan of matrix models to try to integrate multiple effects on different stages. And since we had measured most of the relevant parameters, we worked with Nick Mills to model both the net diet effect and then see how that diet might interact with another mortality factor, such as egg parasitism. The paper we published with Paul Abram put more of a realistic look to what we can expect from a specialist egg parasitoid like Japonicus, but biological control interacts with these other environmental factors. So the bar graph on the upper side is the integrated effect of the diet groups on R0, or the net reproduction, of the Eastern versus Western diet in the absence of parasitism. And you see, even without parasitism, that Western diet causes a 60% reduction in R0. For the parasitism, the relative effect or percent reduction is identical for East and the West, but the apparent effect of parasitism for the West appears to be smaller. And that's just because R0 starts at such a much lower level. So the other insight from these studies is that despite the heavy penalty on nymphal survivorship, the reduction in R0 in the Western group is primarily driven by reductions in female fecundity. So I included this slide in a presentation in 2018 when we were still speculating about how BMSB would fare in an arid climate. And Joanna Fisher's work indicated that high temperatures and low relative humidity can certainly have negative effects on development of BMSB. And of course, that will be helpful, but of course, the climate has this second layer of effects, which is its plant composition. And while we may not be precisely protected by our climate, we may not see those big buildups in wooded areas as they have in the mid-Atlantic, which provides this continuing pressure on agriculture. The unknown here is the relationship of BMSB buildup in residential plantings. On a relative scale, our towns are small and scattered in the landscape compared to, say, Seattle, with long stretches of sagebrush and basalt cliffs in between. These discontinuous landscapes may help or discourage spread or annual migration to crop plants. Conversely, our crop plants will look pretty tasty in comparison to the native plants and perhaps act as a magnet for BMSB in an otherwise unpalatable landscape. And with that, I'd like to thank everybody who contributed to this. And I don't have any polling questions, I believe, Jim, so I can turn that back over to you.
Okay, is Kent
Yep.
on here yet?
Yes, I'm on.
Okay. Kent, how about if you get ready to go here? Our next presentation and the last one during the landscape ecology session is by Kent Dana, talking about BMSB in California, the current status and future outlook.
Great, thanks. Okay, so first I want to thank Joandra Rijal, Joanna Fisher, Frank Zalem, and Judith Stahl. This is their data that I'm presenting. And to talk about California, I'm going to start this presentation with a map, and I'm going to end the presentation with a map. The details of this are not important. Just to show that California's got a diversity of different crop systems, and that diversity is spread throughout the state. And so while we grow a lot of almonds, we don't grow a lot of almonds in Napa County. That's where we grow a lot of grapes. It's a very profitable region for farming. And what I've got here on the lower right is the list of some of the top 10 agricultural commodities in California. And the darker ones, almonds, grapes, strawberries, pistachios, walnuts, tomatoes, stone fruit, are all susceptible to brown marmorated stink bug. And in fact, in today's presentation highlighted here, we touch upon almonds, grapes, pistachios, walnuts, and stone fruit. It was a part of the group's study efforts. We'll start with almonds. So what you see on the ground in the larger picture, those are all almonds that have been dropped. Stink bugs in almonds and pistachios can cause a tremendous amount of damage early in the season by dropping the fruit down to the ground. The pictures on the right are a stink bug on an almond. We see the external damage of gummosis, and we see the internal damage of the spotting or the strikes of feeding from brown marmorated stink bug as we cut into that almond. So just how much damage can a BMSB do to almonds, both in almonds and pistachios? We did cage studies to try to enclose certain numbers of BMSB adults or nymphs on both almonds and pistachios. In this case, they enclosed on 2 varieties, Nonpareil and Monterey almonds. And they did it throughout the season. So they started at early fruit set, and they went until about harvest time, about March through harvest. What we see here are figures from the 2 varieties, Nonpareil and Monterey, Nonpareil on the left, Monterey on the right. The red bars are the infested cages, the blue bars are control. We can see early in the season, week 1, we've got a little bit of natural nut drop, and that happens in pistachios as well. But after that, you can see that the BMSB are causing a tremendous amount of nut drop, about 40 to 98% in Nonpareil and 28 to 96% in Monterey. There is a reduction as the season progresses, and that's as the shell gets a little bit harder, we thought. And it's also probably as the tree sets its fruit load. We switch over now to the not the external damage or the nut drop, but we look at both external and internal damage. We see April, May, June, and July on the graph. We can see in red gumming and in blue the dark spots. So the gumming is the external damage that you see. The dark spots are the internal damage on the nut. And you can see in the set of pictures from April, May, June, and July that we had what seems to be more damage early in the season than later in the season, but we were still getting some dark spots even in July, July feeding. So this can be a season-long pest for almond growers with both damage by dropping the nuts and damage to the kernel directly. Well, the Davis group and Jalandra asked the question, is there a difference amongst cultivars? We've got 4 cultivars here, Tuno, Monterey, Nonpareil, and Butte. And we can see that there's a significant difference in the amount of damaged almonds. And what I've done is I've given orange and red to the 2 cultivars that had the most damage, and 2 shades of blue to the 2 cultivars that had the least damage. They were looking here at shell hardness. Shell hardness decreases from Tuno to Butte, and we didn't see a pattern of damage following this decrease in shell hardness. What I've done next is I've shown their data in terms of the epicarp to kernel thickness, and we can see that in the red arrows on that almond nut in the center of the graphs. When we look at this, the orange and red have got the two lowest levels of damage and the two thinnest epicarp to kernel distances. The Monterey and the nonpareil have got the thickest distance and the least damage. So this is something that they're going to explore a little bit more, that might be an impact on BMSB damage levels. They also looked at walnuts, and we're just showing here control versus enclosed cages, and they didn't see any significant damage on walnuts. That could be a number of different factors, including the shell hardness on walnuts and that distance between the epicarp and the kernel itself. So they're going to follow up on this a little bit more. So almonds, walnuts, two very major crops in California. Another important nut crop is pistachios. UDIT did some work on pistachios, and just like with almonds, we've got in a large picture the epicarp lesion. That's the external damage to the pistachio. We see a clean pistachio nut in the upper right, and we can see a damaged pistachio kernel in the lower left. You can actually make out where and this point it was a stink bug mouth part went through the epicarp into the kernel, causing damage. So very similar to Joanna's work, UDIT enclosed BMSB into cages, and she did it throughout the season at early, mid, and late season periods that matched nut development on the pistachio tree. Added to this, we also compared damage of brown marmorated stink bug to native stink bug and leaf-footed bugs, because they are major pests of pistachio growers in California. And we wanted to find out if BMSB caused more damage than the native stink bugs caused. This is an indication of epicarp lesion per cluster. What we can see is that it's much higher at the beginning of the season, decreases during the season. We've always thought that was due to shell hardness. So similar to what we saw in almonds, there's an increase early in the season to a decrease later in the season. This also follows with the amount of nuts dropped. And in fact, in pistachio, it's not as bad as in almonds in terms of overall damage, because the pistachio tree sets more fruit than it can hold. So there's going to be compensation for natural nut drop and compensation for insect feeding early in the season. But comparing total epicarp lesions towards the end of the season amongst BMSB, the green stink bug, and the leaf-footed bug, we did see that BMSB caused more epicarp lesions overall than the native stink bug and leaf-footed bug. But I want to point out, in the same study, there was no difference in kernel damage at the end of the season amongst any of the large bug pests, BMSB or the native stink bug or leaf-footed bug. What's more interesting to us is a comparison of what we're seeing in pistachios compared to what was seen in almonds. We aren't seeing large numbers of BMSB in pistachio. One of the things we did in a different cage study was to compare the survivorship and development of BMSB down here in Fresno when it's fed on almonds, peach, pistachios, grapes, or a combination of hosts where we gave them all the different combinations of those hosts. What I want to point out, and you can see this is development by different stages, is that on almonds and peach, really there's only about 20% of the population that survives. And in pistachio, it's only about 7% of the population that survives. And none of them survived on grapes. So we see a difference in survivorship depending on the host plant or host material. And that really goes to what Betsy just talked about in the previous presentation. UDIT is also doing a regional development study where we've got BMSB being fed a similar diet in the Fresno area compared to Napa, where we're worried about BMSB damage to grapes, and Alameda, which is basically in the Bay Area. So some very different temperature types. And what we see is that there is a difference in development time. In Fresno, they're developing much, much faster. And we were even wondering about a potential beginning of a third generation that might die out in some of the warmer parts of California. So conclusions to this point, we know BMSB is damaging almonds and pistachios, but not walnuts. We haven't seen any damage to grapes in California yet, especially in the Central Valley. Obviously, they're feeding on peach, and peach was a preferred host in that host plant study that UDIT conducted. But we were curious about why it seemed to be such a major threat to almonds in one study in California, but a minor threat to pistachios in another study in California. And for that, we'll go to some of the work by Joanna and her crew based up in Davis. And she was looking at the impacts of temperature and humidity on brown marmorated stink bug survivorship and development. She parsed this out by looking at different insect life stages of BMSB, 4 different humidities, and 3 different temperatures. And these aren't constant temperatures, but a constant 27°C ambient temperature, and then about a 3 3/4 high temperature of 40 degrees or 42 degrees. The data she had is quite interesting. The first thing we'll point out from her data set is that there was a significant difference between the 27 ambient temperature and the high peak temperature of 40 degrees and 42 degrees for that about 4-hour period, with decreasing survivorship of BMSB with increasing temperatures for those 2 4-hour periods. I want to point out too that there wasn't a lot of difference in the humidity at 27 or 42 degrees, but very clearly at 40 degrees, it appeared that increasing temperature increased survivorship. And she parsed that out even more. And so what we see here is survivorship at the egg, first instar, third instar, and adult stages. And I want to point out here that at the first instar stage, humidity had a significant impact on survivorship, whereas the egg, third, and adult stage, it did not. And this is something that Chuck Engels and I also considered, looking at a dramatic drop in the natural population in Sacramento that occurred at the same time as the second generation eggs were being laid. So I mentioned I was going to begin with a map and end with a map.
Kent, 2 minutes.
Thank you. This is the map from the Crowder group that came out, and it shows BMSB as a pest of almonds. This is our conclusion, but not walnuts. They're looking at the distance to the kernel in almonds as a factor. And on walnuts, the hard shell might have a factor as well. We showed damage to pistachio, but not as preferred as peach or almonds. And Joanna looked at temperature and humidity impacts on BMSB. So we go to the Ilion and Crowder model, and what we see is that, pointing out here, where the almond study was done matches up very well where the model predicted good survivorship of BMSB. Where our pistachio trial was done marked very well an area with poorer survival of BMSB. So it's quite possible, had we worked on almonds in Fresno, we would have had very different results than almonds just 80 miles to 100 miles north of us because of the climate variation in California. So like the map of the different crops, California is probably very different depending on where you are for BMSB survival. And I do have the last slide here. So thank you.
Thank you, Kent. And let's not forget to answer the questions here. Okay, what we have here is about 8 or 10 minutes, 9 minutes, I guess, for question and answer, a discussion session. on anything related to the landscape ecology session. I haven't been able to see the Q&A questions. I'll hear the results.
It seems like most of the questions got answered by colleagues. Does everyone feel like their question was answered adequately? Tom Kuhar may be the only one who wants some more discussion about Catalpa. Tom said he was wondering, hang on a second.
Kent, how did people do on your second question?
They got it right. I was worried that people would not think of it as a multiple choice, but I think it is a multiple choice. I think that host plants, temperature, humidity are all going to impact BMSB survivorship and what's going on. And not only that, you have to think about the microclimate, because pistachios are oftentimes a drier environment, certainly much, much drier than stone fruit. It's very common here to see stone fruit growers with some kind of ground cover understory, where you just almost never see much of an understory in pistachios or almonds, especially late in the season because of their harvest operations.
Hey, Kent, I'll mention, because I think I messed that last question up, my answer. I think when we read multiple choice, I was thinking in my head, it's all of them, but then I'm like, all right, multiple choice, I got to pick one.
Yeah.
So it's possible when you get your results back that I wasn't the only one that messed that up.
Can I ask Kent a question?
Yeah.
Kent, did you guys look at all at reproduction of those populations that you had in those field cages in the different locations? I'm just curious how, you know, if you followed them through to their capacity to lay eggs, because the data are really interesting.
So if we haven't, we will. I think that the first year we did it, there were some issues. Our cages, as we set them up, we didn't realize in the Sacramento site that the food we put in was also great for ants. And once the ants found the food, they found the BMSB eggs. So we can make some tweaks to get that data out as well.
Thank you.
Kent, I have a question also, and it relates to where are most of the pears or peaches grown in California? And are they in an area where you suspect BMSB to become an issue?
So it used to be right around the Fresno area was the key growing area for peaches, peaches and nectarines and plums. And it was also very closely tied into the smaller family farmer. We are seeing stone fruit in California really drop. It's a labor cost issue. So a lot of the crops that are replacing stone fruit are almonds, pistachios, and citrus, which have lower labor costs, especially for harvest and pruning. Peaches, remember, have to be not just pruned every single year, but you've got to thin the crop. There's a lot of hand labor. You've got to oftentimes harvest 3 or 4 times in a season because of differential ripening. Whereas with almonds and pistachios, you go through once with a machine and pretty much you're done. So yeah, Fresno area used to be the most important, and we're getting kind of a movement further north, probably.
Is there anything else for any of the speakers? If not,
Would you like me to show the results from the other
sure.
presentations?
Yeah.
This one's for Ann's.
Yeah.
Any surprises there, or anyone want to comment?
No surprises.
Okay, I'll share, let's see, Dave's now. And that's all of them. So Dave, you have any interpretations of these?
I am curious to know what online decision support systems folks are using outside of the WSU one, which I know really well, because I'd like to look into that more. So if people that answered yes to that, the 42% are willing to send me a chat or something and let me know what system you use, if it's different than WSU DAS or NEWA, I'd love to know what people are using on that front.
You know, it might vary. Everybody may have a different definition of a DAS system.
True.
So
True.
it may be a simple model that somebody's accessing for information too. Okay, what do you say we move on now to the biocontrol session of the meeting? We have 5 different presentations during this meeting, varying from, well, it's all related to biocontrol, but the first one, we're going to talk about Nosema maddoxi, and this is by Ann Hayek at Cornell University. So Ann, why don't you take over, and I'll butt in at 2 minutes left in your presentation.
There we go.
That
Okay.
looks good. Yep,
Okay.
that looks good.
So I'm going to be talking about work done by myself, Carrie Preston in my lab, and Art Agnello. And just so first a little preamble about Nosema maddoxi, because it's pretty different than everything we've been talking about so far. It's a microsporidian species, and up on the top right, those are the spores. They're really small. And they're like slightly smaller than red blood cells. So you really have to have high magnification on a microscope to see them. And down in the lower right, that's the inside of a spore, and that spiral structure actually is the infective structure that's pretty wild, that zooms out of the spore under the right conditions and pierces a gut cell wall. And then all the protoplasm in the spore flows through that tube and into the gut cell. And that's how this infects. And I know that sounds wild, but any of you who've had them in your lab colony know that they're really good at doing this. So this is a Nosema maddoxi. It's a new species described in 2017, but it was first informally described from green stink bugs in Illinois by Joe Maddox in the 1970s. So it's actually a native microsporidia that is from stink bugs, and it has moved over to the brown marmorated. Okay, so first, this was found by Ann Nielsen and her group in New Jersey. And then it was sent by University of Maryland that was having major colony problems with this. And that was followed by Carrie Preston joined my lab and did a survey. And you might have seen this map before. It's part of the publications from her thesis. But the green, basically, what you want to see here is that everywhere that we sampled, we found this microsporidia, which makes sense. It's native, and it's just normally in some of the native stink bugs, and then switches over to brown marmoratids. So first I wanted to talk about transmission before I talk about the seasonality, but vertical transmission is pretty normal for a lot of microsporidia. It means that it's passed from the parent to the offspring, usually from the mother to eggs, either inside the egg, outside the eggs. I've had a couple of undergrads trying to work with this. Carrie did too. And we just haven't proved vertical transmission, but we assume that probably spores are on the egg surfaces. But what about horizontal transmission, which is environmental transmission? And so that's where you'd get transmission within within a field season or in the wintering aggregations. But how does transmission happen? Because these are piercing, sucking mouth part insects, and they're usually feeding on the phloem, xylem, and how are they ingesting the microsporidian spores? Well, they actually feed on feces of other brown marmorated stink bugs. We've seen that happening. They also feed on weakened or dying, recently dead brown marmorated stink bugs, and we've seen them feeding on brown marmorated stink bug eggs. So we think that this is how they're getting this horizontal transmission under field conditions and in your lab colonies. So Carrie did some work looking at the seasonality, and you can see that in 2018, she found levels in springtime at a couple of sites of like 50 and 60% infection, really high levels of infection that then dropped off during the summer and then increased to some extent in the fall at one of the sites. And so it's not constant throughout the year. It seems to be very seasonal, and we are interested in what's going on in spring and fall for why we're seeing the higher levels of infection. And so we have a hypothesis that transmission might be facilitated during the winter aggregations. Bugs are right next to each other, and could one of these horizontal transmissions be happening then? And we actually had some data that supported this for a couple of years. You can see that in fall, there was low levels of infection, and at the same site in the springtime, we had high levels of infection. So that actually fit with our idea that, this is promoting infection in the aggregations. And so many of you, or numerous people, actually sent us samples so that we could test this in the winter of 2019 through 2020. We had fall samples, we had spring samples, and unfortunately, what we found was exactly opposite to our hypothesis, which means that we're back to square one on trying to investigate this and what is actually going on. Because we found more infection in fall than in spring. So this is to be continued. So for Carrie's lab experiments, one of the first things that she did that actually took quite a while, it's not that easy to infect these because you have to get them to ingest the spores. And so we made spore suspensions and then tried to get the brown marmorated stink bugs to drink them, and they wouldn't always drink the whole suspension. And Carrie had a really brilliant idea that had to do with kind of made you think of going to the bar and there's beer, and then they give you peanuts and that makes you buy more beer. And so she offered peanuts, only peanuts, to the brown marmorated stink bugs for 12 to 24 hours before, as she wanted to give them the source spore suspension, and they did much better at drinking the spore suspension. So then for diagnosis, it was interesting. We had 3 different states. Some of them that were, we tried to get them infected, weren't infected, but others, many, we were very successful with these studies. Many had either low density infection, low intensity infections, or high intensity infections. And the impacts that we saw were always higher in for the high intensity infections. Now, I don't know if any of you have actually seen these kinds of symptoms from brown marmorated that you collect in the field, but some of the high infections, we actually saw internal melanization that you could actually see through the cuticle in the abdomens. And we know that that's melanization. There's some kind of immune response going on inside of the insect, and some of the microsporidia are being melanized. And that's what you can see with this arrow in the middle. But I just summarized what we found from all the bioassays that Carrie conducted, that basically, if you give the adult a high dose, they died faster than the controls. They laid fewer eggs, and fewer of the eggs were variable. It had a huge impact. If you gave the adult a lower dose, then we didn't have any significance in survival or the number of eggs, but fewer of the eggs were viable. For the nymphs, we only tested one dose, and it definitely made decreased the survival, and 74% had decreased eclosion to adult. They did not become adults, so it really had an impact on nymphs too. This is all based on lab bioassays with doses in the laboratory, and hasn't been extended to the field, but I think it's a good indication that Nosema maddoxi can have a big impact. So then the last couple of years, one of the things that we did was we got involved. We were always interested in what's the native reservoir that is hosting these spores that, they didn't come out of nowhere in order to infect the brown marmorated stink bugs. And we also were interested in just what was the level of infection that in the Southeast, because our survey didn't have any samples from the Southeast. And so we collaborated with Maribel Portilla at ARS in Stoneville, who was sampling along soybean fields. And she sent us lots of stink bugs, but none of them were brown marmorated stink bugs. And so this was a great opportunity for us to look at the native stink bugs, but not the levels of infection in brown marmorateds. On the table, you can see that we found infections in green stink bugs, brown stink bugs. The Duskies, we didn't get any infected in this study, but we know from a previous that they get infected. Red bandids, really low level infection, and actually in Pedesis also. Some Pedesis were infected. Oops, wrong way, sorry. And Lilac Hong in my lab actually looked at, this infection level of especially the greens and brown and spine shoulder bugs, the Pedesis, in order to see if she could find any, really hot spots from where there was more infection in the counties that were sampled by Maribel's crew. And we didn't find any really good pattern here, but which is something on its own. Okay, so we also did some more host specificity studies, and some of this was with southern green stink bugs that we got from Don Weber because he had a declining lab colony. Yeah. I'm over. It's time?
No, you have 2 minutes left.
Okay, good. I'm almost done. Anyway, the Nazara was infected, and then the harlequin bugs were infected, but we had a hard time infecting them. So just more shows that this has kind of a broader host range. So in summary, it's native to North America, and it shifted onto brown marmorated. It's found in every state we surveyed, but not at every site. And more infection in the fall and spring, and less than in midsummer, and we'll get back to you about that. And the infected adults died sooner and laid fewer viable eggs, and lots of infected nymphs didn't eclose to adults. It has kind of broad host specificity, but especially in the field, the only native stink bug that we ever see infected pretty regularly are green stink bugs. And here is a big list of all the people that have sent us samples, and I want to say thank you. Thank you to everybody. I can't read everybody's names. And thank you to everyone for all of your help. And I have my last slide too.
How are we doing there, David?
We're at about 57% voting completed. We just hit 60.
Okay.
We've been topping off between 60 and 70. So if people could finish up, we'll end the polling in a few seconds and I'll share the results.
So there's a question on the questions and answer. Can I just answer that live?
Yeah.
Okay. So this is a question from David Owens asking if it's possible that Nosema Maddox may be partly behind the population decrease in the East from 2010 to 2012. That's before we started working on this. And so I really don't know how abundant it was at that time. However, I know that after that, remember when I showed you the phenology graph, then we had 50–60% infection at a couple of sites in the springtime. One of those was Greg Kratchik's, one of his sites, and another was Peter Jensch's sites at Highland, New York. And the next summer, they had really low populations. So, we didn't really have a way to quantify density at that point, but we've definitely seen that kind of data to show that it can have an impact.
Okay.
Suggest that it has an impact.
Thanks, Ann. Okay, let's move on. Next, we're going to talk about native natural enemies and their impact of brown marmorated stink bug. And that's going to be presented by Paula Shrewsbury of University of Maryland.
All right, we good? Right screen? Okay.
Yep.
Thanks,
Good.
Jim. Good afternoon, everybody. How you guys doing? Today, I'm going to be talking about what we've learned about native natural enemies as biological control agents for BMSB. And I'll be presenting work that we've done in my lab and also work that others involved in the BMSB SCRI grant have done and shared with me. And the first project I'd like to talk about is a large multi-region, multi-state project with lots of different collaborators that you see here. And the goal of this project was to determine what parasitoids occur or don't occur in the various regions and also in various habitats. And then also to determine the biological control impact of the biological control agents in the different habitats. So here you can see the 5 regions. Oops, sorry. Here you can see 5 regions where the study took place. And here you can see the habitats that we looked at. So we sampled these major habitats, and within each of these major habitats, there are some sub-habitats. So like, for example, forests were mainly wood edges, orchards could be any of these fruits or nuts, semi-natural were campuses, parks, or arboretums, and so forth. The study was done, I'm talking right as the sun's coming in my window. So it's like, so the study was done over 2 years in 2017 and 18. I'm only going to present 2018 data due to time limitations. And I'm going to start by presenting results on the percentage of parasitoids by habitat. So here we have the different habitat types. And all of the results are also being presented by sentinel fresh eggs. So sentinel egg masses were used for sampling in each of these habitat types. And the end value are the number of egg masses that were sampled or go into the data that you're seeing here on the screen. So you can see sampling effort definitely varied. So it was kind of low in the orchards and field crops and riparian, but then you get up to forest or wood edges and it's over 1,200. So keep that in mind when you're looking at the results. What we also have parasitoids represented by different colors. The solid colors are the native parasitoids. And then we also recorded Trissolcus japonicus, the non-native, because that was also emerging from these egg masses. And when we look at parasitoid taxa or species by habitat, we see that Anastatis is common in our semi-natural areas and our wood edges. And we've also collected them from orchards. And in 2017, we also had them in ornamental systems. So they tend to be more of an arboreal type system or an arboreal parasitoid. We also collected Trissolcus in almost all of the habitats except for vegetable field crops. And of those Trissolcus, we only collected native Trissolcus in orchards and non-native Trissolcus or TJ in the riparian. We also collected Telenomus in field and vegetable crops and orchards, and a little bit in wood edges. And then we also had some O. incertus that occurred in wood edges. So what we're seeing here is that specific parasitoids had very specific habitats that they liked. So some of them were more generalist and some of them were more specific. I want to show the parasitoid occurrence or prevalence by region. Okay. And here we see same idea, different number of sampling effort in the different regions. We see Anastatis occurs in the Mid-Atlantic region and in the Southeast region, but we're not collecting it in the other regions. Trissolcus, again, we collect it in all of the regions, except in the Southeast and the West, we only collected native Trissolcus. And in the Great Lakes, we only collected the non-native Trissolcus. And O. incertus we see in the Mid-Atlantic area, but not in the other areas. And Telenomus we see mainly in the Mid-Atlantic and Southeast also, just like Anastatis. So they're very specific, again, on what regions of the country that these parasitoids are occurring in. So in general, certain parasitoid species are prevalent in specific habitats and also in specific regions. Next, I want to talk about the egg fate from these studies. So we determined the mortality factors of all of the eggs in the study. And these are the results of all of the data pooled across region and habitat for 2018. And you can see over 2,700 egg masses went into the data that you're seeing here. And when we look at the outcome or the egg fate, we see that almost 60% of the eggs hatched BMSB. We looked at predation and there was 13% predation, and below that it breaks the predation down into what types of predation it is, complete, incomplete, or stylet sucking, and that suggests what predator probably did that mortality or caused that mortality. Parasitism rates were very low, only 4%, and you can see below the amount by native parasitoids and the amount by the non-native TJ. And then we had this miscellaneous mortality that came to about 23% or so. So you can see differences, so pretty relatively low amounts of parasitism and predation. Then we wanted to look at egg fate by habitat type. So this is pooled across regions. And what we see is that predation is greatest in our wood edges, followed by our row crops. And in orchards, you'll see that there's very little parasitism going on. And that's true also for row crops, little parasitism. But in row crops, we did have these unemerged parasitoids. So it's kind of indirect mortality by the parasitoids. And then in kind of our semi-natural, our wood edges in our ornamental systems, we had similar levels of parasitism. Okay. And then in our riparian area, it was just parasitism by the non-native Trissolcus. So when we consider the impact of the parasitoids and the predators in our different systems, overall, they were low. Parasitoid and predators had low impacts. And then the habitats varied in whether predation and or parasitism was more common in those habitats. Next, I want to talk about a study that was led by Glenn Tillman from USDA ARS in Georgia. And her and her colleagues did a really nice study where they looked at parasitism and predation of sentinel egg masses of BMSB. And they did this in different habitat types. So they've got woodlands, orchards, vegetables, row crops, and vineyards that they sampled in. And what they found was that they identified 10 parasitoids that were attacking the BMSB eggs. And they recorded 2 new records, this Trissolcus psilocysis and basalis. And psilocysis was more common in kind of a tree crops and a little bit in orchards. And then the basalis was more vegetables and row crops. In addition, they found, similar to what we found in the last study, that certain parasitoid species were in certain habitats. So we have the Anastatis and Trissolcus, which is common in our more arboreal type of habitats. And then Oencertus and Telenomus are kind of more across the board as far as what type of habitats you find them in. They also looked at predation and parasitism rates in their studies. And here they found that in soybean, and this was the same for cotton, that predation was consistently high at pretty consistently high levels in their crop, in the soybean and corn and cotton, whereas parasitism was quite low. They also looked at predation and parasitism in apples and on the wood borders of apples. And here you can see that parasitism of the apples and on the wood edge was quite high in June and up over 70% in the apples. So that was nice. And then this went down a little bit over time. They had a little bit more predation in the other habitats too, and parasitism. So again, variation over time in the different types of habitats. One of the other things that Glenn and her colleagues pointed out was that when you use sentinel eggs in your studies, that you are, oops, sorry. There we go. When you use sentinel eggs in your study, you are overestimating predation and underestimating parasitism. So they compared predation and parasitism from sentinel eggs and from naturally laid eggs. And what they found is if you look at predation from sentinel eggs, it was not quite twice as high as predation from natural eggs. So they felt they were overestimating predation. And if you look at parasitism, on sentinel eggs, it was 12%. On natural eggs, it was 26%. So over twice as much. And this is supported by data that we have from Maryland that we did several years ago in 2012 and 13 in ornamental tree nurseries, where we compared parasitism rates between sentinel egg masses and naturally laid egg masses. And you can see here that sentinel egg masses dramatically underestimated the parasitism that occurred on the naturally laid egg masses. So what does this mean? It means that we should try to use naturally laid egg masses as much as possible, but as researchers, we all know that that's really hard to do. So we need to consider these results when interpreting our data. So this under and overestimating of our data. So what we've been seeing so far in this data is relatively low levels of parasitism and predation. And though that varies from habitat to habitat and in different times, but in general, the numbers are low. So the idea now is to try to focus on ways to try to increase or enhance the impacts that these natural enemies, these native natural enemies have on BMSB. And so right now we've got a study underway, an augmentative release study using Anastatis reduvii, and we've picked this native parasitoid because it's one of the most prevalent and abundant parasitoids in our arboreal systems. And it's had the biggest impact as far as the native natural enemies go in many of the arboreal systems. And we want to examine if augmentative releases of this parasitoid can increase biological control impacts. And we're doing this study in North Carolina with Emily and Jim, and then in Maryland, my lab and my research associate Alina are working on this. We're only in the first year of the study so far. We just finished it. But the study consists of 2 pairs of study sites in each state, a release site and a non-release site. And we're doing releases in wood borders, and the wood borders can border fields, crops, orchards, or unmanaged areas. And we have a release tree or a non-release tree. And then out from distances on both sides of that, we are sampling with sentinel egg masses and with yellow and green sticky cards. And these are results, excuse me, from the first year in North Carolina. And these aren't results for Anastatis reduvii because the numbers were so low that we really they couldn't pull much out of it. So instead, we're at least looking at the amount of all the parasitoids. So parasitism rates and the number of parasitoids in these different sticky cards, colored sticky cards. So here we have release and non-release wood borders, wood edges bordering apples, and release and non-release wood borders on managed areas. And you can see what we found was that pre-release, and we have pre-release and post-release counts. So pre-release totals egg parasitism was higher than post-release, and that's probably due to seasonal differences. And there were differences in locations between the pre-release and the post-release. And we see similar results when we look at the abundance of parasitoids from the sticky cards. Another study that I want to mention is looking at another way to try to enhance natural enemy populations especially with the Anastatis reduvii. And my graduate student, Maddie Potter, is working on this. And we're trying to identify alternate in-season and overwintering insects for our Anastatis reduvii. Anastatis is a generalist egg parasitoid, so it attacks other types of eggs. And then also identify host plants, which A. reduvii is commonly associated with.
2 minutes.
Okay, thanks. So Maddie's put together a citizen science project where she's recruited about 50 master gardeners from 5 different counties in Maryland. The idea is that these master gardeners will go out and collect eggs from a big diversity of areas in our state. They ship the eggs to Maddie, and then she'll identify what the eggs are, what natural enemies emerge from it, and then what host plants they were collected from. So we have some preliminary data on this. If you go to the literature, it documents alternative hosts for Anastatis, and you can see there are a number of them here. And the 2 in green were new records of Anastatis reduvii emerging from these hosts, at least from our knowledge. We haven't found this in any of the literature. So Carolina mantis and leaf-footed bugs. So you can see a whole diversity of insects across orders for this. She's also doing a meta-analysis looking at the host plant data to try to determine which plants are closely associated with Anastatis reduvii, and then identify factors that might let us know what is it about those plants that favor Anastatis. And these are some of the plants that we've already sampled Anastatis from. Another study, we know that there's Trisulcus japonicus out there, the non-native and our native parasitoids. And we're looking at some competition between those. I don't have time to present those data, but my research associate, Lena, is presenting this at the Eastern Branch meeting. So if you want to try to catch that talk, that would be pretty interesting. So overall, what's the outlook? We have low levels of predation and parasitism by habitat type, and we have the differences in region. We also know that the overall impact of our parasitoids and predators is low. So what do we have to do? I think that means that now we need to focus our research on trying to figure out ways to implement and enhance those natural enemy populations in ways like we've started already with the augmentative releases and looking at ways to modify habitats to favor those native natural enemies. And I think that's it. With that, I will put up my last slide.
Okay. Thank you, Paula.
Welcome.
Now we're going to continue on with biological control, but the last 3 presentations of the day are going to be related to Trisulcus japonicus, which is the first presentation is going to be by Nicole Quinn and along with Chris Berg. Nicole will be giving this presentation. Nicole, hopefully you're on the line.
Yep, yeah, I'm here.
Great.
Okay.
I'm sorry, I didn't read the title of your presentation, which is.
Oh, it's okay. They'll see it in a second.
Okay, fantastic.
Okay, does that, everyone can see that?
Yes.
Yep, looks good.
Okay. Good. Okay. Yeah, so I'm presenting this on behalf of Chris Berg. So I was his PhD student up until December 2019. So half of this presentation will be things related to my dissertation, and the other half will be some of the newer stuff from his master's student, Jared Dyer. So should be a good mix of things. So Trisulcus japonicus is a parasitoid brown marmorated stink bug that I think most of us in this group are familiar with at this point. And it's one of the most effective natural enemies of brown marmorated stink bug. And it was found as an adventive parasitoid in Beltsville, Maryland in 2014. It was the first detection in the US. And then it was subsequently detected in Frederick County, Virginia, which is where the research I'll describe takes place. And it wasn't detected anywhere else in Virginia at the time. but that's since changed a little bit. And Trisulcus japonicus has actually been found to be spreading both on the East Coast and the West Coast, a few more states every year, it seems. And so this map just shows the Trisulcus japonicus detection zone locations as the little dots overlaid over the, the classic brown marmorated stink bug map that we all know and love. So basically one of the main goal of Chris's lab is to optimize the efficiency, effectiveness, and the efficiency and effectiveness of TJ surveillance efforts to track the current distribution, range expansion, and changes in abundance over time in Virginia. That's at least one of his, the lab's main areas. So to that end, from 2016 through 2017, we determined the distribution, or we explored the distribution of BMSB, its egg masses and parasitoids in host trees, so individual host trees. And then in 2016, we also started using yellow sticky traps. We piloted using those to see if we could effectively use them to catch Trisulcus japonicus, and it proved to be an effective method. And then in 2017, we compared yellow sticky traps to sentinel eggs to see, if there are any differences in their efficacy in detecting Trisulcus japonicus. And then from there, we went into some kind of deeper questions about the spatial and temporal effects of TJ distribution over time and among different habitats and host plants. And since 2019, he's been looking at the effects of different olfactory stimuli on BMSB egg masses, whether or not they're laid in trees, and also whether TJ is present. So to recap some of the things that were found in my dissertation work, which was that there were, when you put up traps in Tree of Heaven, which is one of the most abundant BMSB host trees in the landscape in Winchester, Virginia, where this work took place. If you put them at the lower part of the tree, the mid canopy and then the upper canopy, we found that the mean weekly captures of adult BMSB across both years were highest at the top of the tree compared to other trapping locations. And we also found something similar for nymphs, although not as strong. And you'll see that there's, like the previous speakers have talked about, the number of BMSB that you find in a given year can vary quite a bit, which is why it's important to do more than 1 year, as we did in this case. But we found similar trends among years, which was good, or between years. And then to further look at this, we cut down Tree of Heaven in just on the edge of different orchards in the area to see if there were any differences among heights within the tree of where you would find BMSB egg masses and Trisulcus japonicus. And what we found was that the most eggs were found, or egg masses were found in the mid canopy of the tree compared to other locations. And that's also where we found the most Trisulcus japonicus. When we didn't find any parasitized egg masses in the lower portion of the tree, even though there were egg masses present. And so all of that's very labor intensive, producing the sentinel egg masses, and then also cutting down trees obviously is not pragmatic for widespread sampling or widespread detection and monitoring. So we decided to test out yellow sticky traps and see if those were effective for detecting Trisulcus japonicus. And the good news is that it was effective, and it takes a lot less time. You can't ask the same sort of questions as with a sentinel egg mass, or you don't get as much information about predation and things like that, but it's a tool that's available that can be helpful for certain things. And so using yellow sticky traps, we decided to look at different habitat types in the Winchester area, Frederick County area, and see if there were any differences in the likelihood of Trisulcus japonicus detection in different sized habitats. So we had basically 3 different sized habitat patches. So we had basically a small island. So it was just a little tiny patch of Tree of Heaven. We had a windbreak treatment that was kind of like a intermediate size, like long, skinny, like a windbreak that's like on the edge of a orchard or field. And then we also had a woodlot treatment, which was, a bigger, more contiguous piece of wood. And we would deploy yellow sticky traps to the edge of those and see if there were any differences in captures among them. And what we found in 2018 was that the windbreak treatment, so kind of the mid-sized treatment, was there were significantly more detections of Trisulcus japonicus there than in the woods treatment, so the bigger, more contiguous one. However, that those results weren't repeated in the subsequent year. So it can be quite variable. So that just shows you, the importance again of doing multiple years of sampling. So I think it'd be interesting to see trends over time and see if, whether there is a habitat association. But our results were kind of inconclusive in that sense. And using those data, we also tracked the the detection of Tricyclus japonicus over time. So we started sampling in mid-May and then ended sampling at the end of September over a 2-year period. And the total captures per week of Tricyclus japonicus lined up fairly well, with first captures in early or mid-May-ish. So, May 18th and May 13th were the 2 dates. And then the first peak in Tricyclus japonicus captures would be, mid-July or so, or towards the end of July. And with a second peak around, mid to or early August. And then the final detection of Tricyclus japonicus occurring in mid to late September. So those results lined up fairly well between years, which also tracks decently well with the phenology of BMSB. And so then we decided to see if there were any effects of the type of tree that you put the sticky trap in on the detection of Tricyclus japonicus. So to that end, we deployed yellow sticky traps in 2 different several different combinations of pairs of host plants. So it'd be tree of heaven and one other host, like black locust or black walnut, for example. And we wanted to see if there were any differences. And so we had them paired so that they were between each deployment location, or within a pair, the traps were between 10 to 25 meters apart. So they were close, but not on top of each other, because remember, these are, it's a visual attractant, so you don't want them directly competing. So what we found was that there were no significant differences among these pairs, or among these hosts in Tricyclus japonicus captures in both years. So it seemed like that you were just as likely or not likely in 2018's case to capture Tricyclus japonicus. But then we repeated it in 2019 with a slightly different design, in that we found, based on our windbreak, our habitat study, which found that windbreaks might be one of the more effective places to deploy yellow sticky traps for detecting Tricyclus japonicus. Based on that, we located more windbreaks that had several different host plants of BMSB in them. And what we found was that if we deploy yellow sticky traps in those host plants, that there were significant differences. So in this case, there were significantly more Tricyclus japonicus captured in black locust compared to tree of heaven, which tree of heaven has been our main was the main host that we were looking at for all of the studies. So it's kind of an interesting thought. Maybe it's important to include a diversity of hosts when you're doing these sampling efforts. And then we also kept track of the other parasitoids that were captured on these yellow sticky traps. So all of these are parasitoids of importance to brown marmorated stink bug biocontrol. And Tricyclus japonicus is the blue slice of these pies, and or the darker blue one, I guess. And it was definitely the dominant parasitoid in both years, but there's still a healthy diversity of other parasitoids. And so now we're kind of moving into Chris and Jared's work. So again, I wasn't directly involved in any of this. So if you have any detailed questions about any of this, definitely ask them and not me, because I was not there. So basically, they wanted to see the effects of different olfactory stimuli on BMSB and Tricyclus japonicus. So the likelihood of whether or not you're going to detect them using a variety of different sampling methods. And so the first thing they did was they deployed a BMSB lure on a tree of heaven, and just wanted to see if the BMSB egg masses were more abundant in trees that had a pheromone lure in them. They also deployed pheromone lures in combination with a sticky trap to see if you could catch more TJ on traps that had that, or they also had a combination where it was a BMSB egg mass on Tricyclus japonicus, or on a yellow sticky trap, and they compared that to just a regular sticky trap without an egg mass to see if there are differences in Tricyclus japonicus captures on those. So with that, we'll kind of get into a summary of that, which is essentially there was no effect of deploying pheromone lures on trees on the number of egg masses that were within that tree. So they deployed the pheromone lure in the tree for 2 weeks, and then they cut the trees down, and they found that there were no additional egg masses on there compared to a control tree. And then there also wasn't an effect of the pheromone lure on Trisulcus japonicus captures. And they also, so they found, to move this window out of the way, they also found no effect of the fresh egg masses on Trisulcus japonicus captures compared to just a control that just has pieces of leaf on them that had, the leaf pieces had been exposed to gravid female BMSB, but did not have any eggs on them. So there's no effect there. And there was also no effect of the BMSB lure on captures of Trisulcus japonicus. And they also decided to look into the trap location again, so kind of look at it in a little bit of a different way, where they deployed the yellow sticky traps either at the lower canopy location, so kind of close to the ground, or at mid canopy location. And what they found was that at least some sites, the lower canopy location, there were significantly more captures of Trisulcus japonicus compared to the mid canopy location. But that was site specific. So they're going to be repeating that and seeing what happens. And so they also found a positive relationship between captures in the mid canopy of Trisulcus japonicus and captures in the lower canopy of Trisulcus japonicus. And so all of that will be repeated, like I said. And so they also wanted to see if there was a relationship between Trisulcus japonicus Trisulcus japonicus detection and BMSB captures. So to do that, what they did was they deployed yellow sticky traps near clear sticky traps that have the BMSB dual lure on them.
2 minutes, Nicole.
Yep. And so what they found was that the captures of adult BMSB and Trisulcus japonicus tracked fairly closely over time. And so that the gray line is the nymph, second instar nymph BMSB captures. So the black line is the adult BMSB captures and red is TJ. So you can see they tracked fairly closely. And what they found was that there was a significant positive relationship between TJ captures and BMSB captures for adults and nymphs, with a stronger relationship for nymphs. And so they'll be repeating that this year with more sites. And they similarly found that TJ was the dominant parasitoid compared to the other native parasitoids. And that's a lot of people to thank, a lot of people to thank to make all of this work happen. And that's it.
Thank you, Nicole.
Yep, you're welcome.
Okay, we're going to continue with Trisulcus japonicus. The next presentation is by Joe Kaiser, redistribution of T. japonicus and effects on infochemicals on stink bug parasitism.
Okay.
So Joe,
Yeah,
like everybody else, I'll let you know when 2 minutes is up.
Okay. Can you see it all right?
Yep, looks good.
Okay. Yeah, so I'm going to talk first a bit about some of the redistribution efforts that we've been doing, focusing on Delaware, and then kind of transition into some of the work we've been doing with pheromones and the effects on Trisulcus japonicus foraging in the field. So yep, this is a map that you have seen a few times. And you know, what I want you to just notice here is that Trisulcus japonicus continues to spread geographically, and it tends to stick around once it gets to a certain location. So it seems to get well established fairly easily. So it's across a broad geographic range, but in a lot of areas, these local population densities are still quite low. They're they're variable, and in some places, particularly on the West Coast, we're starting to see higher densities and higher parasitism rates, but they can remain quite low in many locations. So redistribution efforts can help to increase the spread of this parasitoid, increase the local density, and hopefully increase the biological control impact. So one of the questions we were asking is, what is the optimal release strategy? And so we're kind of using this as a way to also study some of the colonization processes for Trisulcus japonicus as it shows up in new areas. So this is one study that we conducted in Delaware, where we had 3 treatments. We had these high release areas where we would release about 400 wasps at 2 different time points in the season. And that was using the Nielsen phenology model to kind of match with peak BMSB oviposition events. So we do this twice per season for a total of 800 in the high release, about 80, so 2 times 40 in the low release, and then controls where we didn't release any wasps. And we did this throughout the state. And so these were spatially blocked with 2 kilometers between each of the release locations. And so all of our sites were forested habitat edges. So in our first year, it was we got fairly low recapture, but it was trending in the direction that we predicted, with the high releases having slightly more detection and none in the controls. But the subsequent year, that kind of just all disappeared. And in neither year was there a significant effect of treatment. This year, due to the pandemic, we had to limit our monitoring efforts, but we have deployed yellow sticky cars in those areas, and we're processing those results. So we're going to continue to monitor, and we may start to see separation in treatments in the future. But over these first 2 years, it looks like these parasitoids aren't sticking around. If they are, and it's unclear if they're dying out or if they're just dispersing widely. And there have been a lot of other efforts to redistribute this parasitoid, and which I just don't have time to go into all the other efforts, though several people have spoken already on their local releases. So one thing that we did, this was towards the end of 2019, is we were thinking about why this parasitoid isn't building up locally, even at our release sites. And so we thought dispersal may have something to do with this, that the parasitoid may just leave the area so quickly that we can't redetect it. So this was just a kind of a one-off study we did, where we set up these approximately 2-meter tall potted maple trees, and we affixed egg masses to the leaves and released a bunch of wasps in the middle. So for this time we did, there were 1,700 female adult wasps released. And so just what you can see here is the red dots indicate trees where we had parasitized egg masses by Trisulcus japonicus. One thing that I didn't mention is that we alternated treatments for these trees, where we would bag a single branch and put BMSB adults in that bag to walk around on the leaves. We did that for some of them, and some of them we bagged and left and put no BMSB in there. And so we were trying to see if the prior presence of BMSB had any impact on the ability of the parasitoid to detect and then parasitize the egg masses there. And this was, just a one-off kind of little pilot study, and it was trending in this direction where there was some impact of caramone, the caramones left behind. So we wanted to pursue that a little bit further. But one thing that we kind of take home also from this is that the parasitoids, for the most part, we did get 26% parasitism of the egg masses in the area. But if you think about that by all total, if a single female is parasitizing each egg mass, then that's about 0.7% of all the females actually parasitize something locally in that time period. We believe that they are dispersing quite a bit, but there's still a lot of work to be done on that front. In terms of the caramone work, this was all based on some work that Sean Boyle had done during a master's program at University of Delaware. And then Robert Malik followed up on a lot of that. And so what I'm showing here is a paper that Malik et al. It's in revision right now in biological control showed. So this is laboratory work where we would have adult or nymphal BMSB in the red, or a non-target Podiasis maculoventris in green here, or a control where there's no exposure to filter paper by these stink bugs. And then we would place it under this camera and track it with EthoVision software and see how the wasps behave differently. And what you see here, this is just showing retention time, so or residence time, the amount of time that the parasitoid spent on that filter paper. And if a BMSB stink bug, adult or nymph, had been present previously, the parasitoid spent a lot longer there. And there's sort of an intermediate effect with the non-target Podiasis, which correlates with what we would see in our host range tests as well, where Podiasis maculoventris is attacked by Trisulcus japonicus, but at an intermediate rate. So we wanted to see, do these lab results mean anything for field level parasitism? So I'm just going to quickly go through 4 experiments we did. They all have the same basic design. So we reused, this past summer, reused those maple trees, and we set them up in circles. So there were 3 different circles, 3 spatial blocks. And each block had 16 trees. There's a 10-meter radius there and about 3 meters between each tree. We'll treat each tree as an experimental unit. And so what we were able to do is apply different treatments to these trees and just mass release a bunch of wasps in the middle of each circle. And this would just sort of increase our likelihood of parasitism. And, we hope that would have a better chance of actually seeing an effect of the treatments. And so each time, depending on availability, we release anywhere between 200 to actually sometimes over 2,000 females per release. And so while this wasn't part of a redistribution project per se, we did end up releasing about 20,000 wasps right on the University of Delaware campus this past summer. So we are putting them out there. So the first experiment, what we did is similar to the experiment the previous year. We bagged a branch, and we would put 4 BMSB adult females on a bagged treatment, or we would put none in there for a control. We would leave them for 48 hours. After that, the bags and the bugs were removed, and we would release the wasps. And then we collect them after 5 days. And so what we saw was there was a really clear effect of prior BMSB exposure. So this kind of fits with the lab results. It's pretty large difference, not quite double. In some cases, in some of the blocks, though, we would have double the parasitism or more if a BMSB had been present. There didn't seem to be any impact on native parasitism, though. So in the second experiment, we wanted to see if there would be a similar effect with Podiasis maculoventris, because there was that intermediate effect in the lab. So we did the same thing, but this time using Podiasis, our native stink bug. And what we saw there was that there was no effect. It's slightly lower replication, but still pretty good. Yeah, it was overall much lower parasitism, and no significant difference between the treatments. So that's really positive in terms of non-target risk, that sort of our expectations from the lab that the ecological host range may be a bit lower because for the non-target, because the non-target is harder to detect for the parasitoid, that the ecological host range might be a bit more limited compared to what we were seeing in those initial host range tests in the lab, where we just kind of forced the wasp on the egg mass. So then the next 2 experiments I'm going to present, we want to look at the effect of the synthetic caramel. So Nicole just talked a little bit about this work that Jared was using. One of the components that have been identified as kind of the main components of this caramel. There's two components that we've identified, the E2-decenol and N-tridecane. So in the previous, what Nicole was presenting, they were looking at N-tridecane. And so in our these next 2 experiments, there's no sort of logical reason for having them in 2 separate experiments. It was more just logistical and what we were able to do. So The first one, we had 3 treatments where we would apply a solution to a filter paper and place an egg mass. These are all fresh egg masses for all of these experiments, by the way. We would place that in the middle of the filter paper and then affix that to the tree. And so for the first treatment, we had just N-triticane alone. So from our previous work that Robert led, we found that N-triticane had an attractive effect when applied as a synthetic in a hexane solution. And a ratio where you have 9:1 triticane to decenol also had an attractive effect. And then, so we wanted to see if there was any difference between those two. And then we had our hexane control. And in our release, we did not see any significant impact. They kind of trend, that the control's lower, but there's no impact there that we were able to see. And I'll talk a little bit about why I expect that is, why I think that is the case after talking about experiment 4. And this is, so in this case, we just wanted to look at N-triticane alone again, but now comparing it to the E2 decenol alone. And the reason we wanted to look at this one alone is that in our previous work, it seemed that when you had E2 decenol alone, it had somewhat of a repellent effect. So this could be useful if there's any case we want to kind of keep the parasitoids away. That's something to look into in the future. But we wanted to look at these 2 and then again a hexane control. But again, we saw no effect. So with both of our experiments using the synthetic components of the caramone, we didn't see any significant effect of treatment. As a final thing, this was done at the Beltsville lab with Don Weber, and Megan Herlihy did most of this work. This was just to look at, okay, we're mass releasing these parasitoids in the previous experiment and kind of an artificial field experiment. But just looking at ambient field populations of parasitoids, do you have any effect of on parasitism by using some of these treatments? So we did look in the field. This was in Maryland. Again, at a 9:1 ratio of triticane to decenol, a triticane only treatment, and then a hexane control. And so this was on 10 different landscape trees blocked at 4 dates. And so in this case, it was only frozen egg masses. And they were placed for 72 hours out in the field. And what we see is, again, no effective treatment. And there was, quite a bit of replication here. And there was quite high parasitism when you add in native parasitism. Fairly low Trisulcus japonicus parasitism, though. And yeah, again, no effective treatment. So this is likely because both of those compounds, the decenol and triticane, are highly volatile. And so we did an experiment on the side to look at on these filter papers at kind of time 0, time 1, 2, and 4 days out. How quickly does this stuff volatilize? And after 2 days, there's basically none of the compounds still present. And after 1 day, it's already very diluted. So it's possible that there's maybe some trace other chemicals that are deposited by the stink bugs that make these particular key compounds not volatilize as quickly. It may have something to do when it's deposited on a leaf surface or something else. So anyways, in conclusion, prior exposure of BMSB did increase foraging efficiency and parasitism rate of eggs significantly. But this was not the case with podiasis, so that's really good. And those results are consistent with our initial conclusions about reduced non-target risk from the lab. Application and the kind of precise composition of these synthetic caramones for use needs further research. And one question is, how in the field, how is the wasp actually responding and detecting these chemicals? Are they detecting them as a volatile attractant or as some kind of contact pheromone? our lab work, just sort of anecdotally, it definitely seems to be a contact hormone where you see the change in behavior upon contact. However, some other researchers have found that you can get an effect of these in a Y tube setting, so they can detect them as a volatile as well. So it's unclear how they're mainly used though in the field. And thank you.
So we're going to end up the session today on biological control with Kim Holmer talking about the status of T. japonicus in the US. He kept this a really simple title, so we'll see if his talk is that simple.
Okay. Can you see my title slide now?
Yeah, we can see them all.
Okay.
So I'll let you know when you have 2 minutes left.
Okay. All right. So you've heard a lot already about Trissolcus japonicus from Nicole and Joe's talks. There's a lot more that's been done, which I'm going to try to summarize. And most of the people on this list of collaborating institutions have people in laboratories that have contributed to some of this work on Trissolcus japonicus. So I'm going to try to cover very briefly a couple of, sort of abrupt things that Joe and Nicole didn't cover in their talks. The status of the advent of populations in the US, redistribution efforts, some of which has been touched on by some of the speakers already. Opportunities to conserve Trissolcus japonicus populations. Whoops. That wasn't supposed to happen. The status of the petition for field release and where we might go on. Now, as various speakers have alluded to, the COVID situation has prevented a lot of field activities, and I think that's probably contributed to the fact that we don't have a lot of new survey information, and there are no new reports of Trissolcus japonicus in new states since 2019. I think COVID probably has contributed to that. The map in the upper corner shows in the green the most recent reports in Utah, California, and Michigan, and also over the border to the north in Canada and Ontario and British Columbia. I suspect probably we could find it in some other states, but more surveys will be needed for that. Samples from many of these locations have been analyzed, and so far the results are the same as in previous years. We know from our studies of Trissolcus japonicus in the native range that there are, according to CO1 barcoding gene, there are 6 major lineages that occur in different parts. And the largest lineage is widely distributed throughout the native range, lineage 6. And all of the adventive populations in North America, as well as the adventive populations in Europe, in Italy and Switzerland, belong to this lineage 6. So there's quite a bit of diversity potentially within that. Even though they come from the same lineage, the same matrilineal lineage, there is still substantial genetic variation that can be picked apart with other tools such as microsatellites. And we know from the previous studies, the previous analyses of the populations in North America that our adventive populations fell into 3 distinct groups, which is still the case. And the populations in Italy and Switzerland are completely different from the ones that we have in the US. So the thinking is that these probably all represent separate, distinct introduction events. We don't know yet how or where exactly they came from, but there are still a couple of the new populations that haven't been analyzed to put into one of these groups. Now, very briefly, a lot of information I've received from the states where redistribution events and surveys have been conducted, but I wanted to point out some of the highlights from some of these areas that I think are the most important takeaways. In Michigan, for example, they've been releasing now for 2 years at 16 different sites, and they've already gotten recoveries at 4 sites. The recoveries are at low rates, but they indicate that there's been overwintering, local reproduction, dispersal from the release sites and summer activity, which is an important takeaway. In Ohio, Celeste Welty's group initially found them in 2017, I believe, at the OSU Research Farm. And in subsequent years, they've made releases at 10 commercial farms. And although they haven't made any recoveries of Trisulcus japonicus at the commercial sites, they've repeatedly recovered the Trisulcus japonicus in good numbers at the research farm where it was first found, and they found them every year. So it's clearly established well at that location. In New York, data from Peter Jensch and Art Agnello indicated that they've been making releases for several years, starting out with parasitized egg masses, then moving on to releasing adults. And they have overwintering recaptures at 13 of 14 sites where previous years releases were made. Also a good sign that there are established populations in some of those areas in New York. And I have a map in a moment to show where those are. In Washington, Betsy mentioned that they've been making releases across the state throughout this year, although they haven't had an opportunity to do surveys, but they've tried to focus these releases near population centers to give them a better chance of establishing in diverse habitats. And Josh Milne has also made releases in the northern part of the state. Josh's releases are not shown on the previous chart, but they're up here in these two northern counties. Can't get my cursor to move here. The yellow stars show where Betsy's releases were made, and you can see that they're distributed across the state. So there's pretty good coverage in Washington. And of course, Washington is the first state on the West Coast that detected Trisulcus japonicus. In Oregon, we have these charts sent from Claire Donahue and Nick Wyman's lab. And the two maps in green and lavender show the recoveries that they've made from sentinel and from wild BMSB egg masses at the top, and from yellow sticky card traps that they've used to search for or to survey for Trisulcus japonicus in the lavender map. And you can see that they pretty consistently have a lot of recoveries at many sites. And the interesting thing that I wanted to point out here is that as you see in the yellow box, they have pretty significant levels of parasitism in the sentinel and the wild BMSB egg masses that they've collected. So that's a good sign for establishment in that area of Western Oregon. Diane covered a lot of this information already in her summary of the situation in Utah, but I wanted to point out for the Utah results that the recoveries show that Trisulcus japonicus has successfully overwintered at several sites between 2019 to 2020. And you can see from the map, the yellow and the orange and the red recovery sites, that they're fairly widely distributed throughout central Utah. And they're getting significant levels of parasitism in their sentinel egg masses. 47% was the report on the slide that I got. So again, very significant levels. Now here we go to New York, and this slide from Peter shows the two areas in the Hudson River Valley and the Great Lakes area up around Lake Erie. No, that's Lake Ontario, where they've made a lot of releases. And the large circles within my yellow highlights show the 14 sites where they have made overwintering recaptures. So there's fairly good distribution throughout New York now as well. Chris Berg's releases throughout Virginia are shown on this site. The red dot shows the location of his lab in Winchester, where a lot of the studies that Nicole talked about were based, and where they have the largest population of Trisulcus japonicus. They've been capturing large numbers repeatedly since 2015, as you can see in the diagram throughout the season. They've gotten very, very good numbers. I think on Nicole's slide, they've last year, I think she mentioned over 1,000 recaptures of Trisulcus japonicus and through various studies. Now, over the past couple of years, Chris has also been releasing at a variety of other sites up and down Virginia. And you can see the small red icons here and there, and the numbers in white show the numbers of recoveries that he's gotten from those releases. So not very many recoveries so far for the releases made, but he is getting them at some of the sites now. In Pennsylvania, the takeaway here from this slide provided by Hillary and Greg show that they've been using yellow sticky cards to monitor for Trisocus japonicus, and they find that the yellow sticky cards show that it's widely distributed throughout the state. And what's really interesting here is that the only place they made releases of Trisocus japonicus was in the site represented by red in Lancaster and southeastern Pennsylvania. All of the rest of the sites are natural spread. Very encouraging. And in Ohio, as I mentioned earlier briefly, that Celeste's group has been finding no recoveries of Trisocus japonicus at the commercial farms where they've released, but they're repeatedly getting recoveries throughout the season. And the chart with the red oval here shows the recoveries of egg masses. The ones in red show the ones that are parasitized by Trisocus japonicus throughout the season. So you can see that it's not just a few random recoveries, but regular populations that they're finding at that site. And in, I think, the most recent year, they've had recoveries or parasitism rates as high as 41%. So we're starting to see significant levels at some populations. So in summary, the distributions, even though we're not seeing them move as fast as we would like, or in large, recover them in numbers as large as we would like at some sites, at this point, we are seeing them to be fairly widely distributed, apparently still moving into new areas and showing signs of having potentially significant levels of parasitism that could show eventual significant contribution to mortality of the regional stink bug populations. And they're overwintering successful at these locations. So these are all good things that point to potentially promising things from Trisocus japonicus as we move forward. Now, one of the handicaps in conducting field work has been the availability of Trisocus japonicus. In order to have Trisocus japonicus in significant numbers to rear, people have had to maintain large healthy colonies of brown marmorated stink bug, and that's expensive in terms of research space and investigator time. And as Anne mentioned, a lot of people have had problems with Nosema contamination and so on. So there's been a lot of important work done in the last couple of years on methods for optimizing the production and the use of stink bugs for production, mass production and rearing of Trisocus japonicus for these field studies and redistributions. So this kind of work should make it much easier for researchers to continue this work going forward. Now I'd like to say a few words about opportunities for conserving Trisocus japonicus in IPM programs. Of course, one of our major goals is to have Trisocus japonicus operate in the background in the landscape on its own. But in the real world, we also want them to be able to move into grower situations and impact populations and crops, if at all possible. And there are many opportunities for combining biocontrol with various kinds of semiochemically based and other behaviorally based methods for managing stink bug populations. And this chart just shows some of the range of things. And several of the speakers tomorrow are going to talk in more detail about some of these opportunities. I just want to mention very briefly how Trisocus japonicus can fit into some of these opportunities. Tracy's lab, for example, in conjunction with a lot of other opportunities at other locations, have been investigating opportunities for integrating Trisocus populations with various methods of reduced pesticide applications to help conserve populations of Trisocus japonicus that can move into the crops or operate in the immediate habitat. Some of this work has just been published in Insects in this publication by Dalton Ludwig and all.
Give him 2 minutes.
Tracy will talk more about this tomorrow, I'm sure. Also, Ann Nielsen's lab has done some work looking at the impact of insecticide drift onto surrounding habitats. And she did an experiment, or her lab did an experiment looking at whether or not the drift onto neighboring plantings of buckwheat significantly impacted the populations. And preliminary data was encouraging because it showed that there was no difference in survival between untreated buckwheat and buckwheat that may have had some drift of pesticide from the peaches that were treated nearby. So again, very encouraging. There's been some nice work done with the contribution of floral resources to help conserve Trissolcus japonicus populations. And some nice work has been published by several groups, including Jana Lee's lab and Glenn Tillman's lab, showing how some of these plant resources can help contribute to increased longevity and vigor of Trissolcus japonicus females. Next effort is to try to move this kind of research into the field to see whether or not the impact of these resources can be seen in actual plantings. And some efforts have been made in this direction. So far, the results are preliminary, and the low recovery rates have confounded trying to determine what kind of impact this can contribute, but more research needs to be conducted in this area. And finally, say just a few words about the hopes for a petition for field release. And of course, as we've talked about in previous meetings, one of the most important aspects in getting permission for field release is the ability to demonstrate non-significant impact on non-targets. And we now know from various field studies and molecular technology that Trissolcus japonicus and many other parasitoids are generally not purely host specific. We know that Trissolcus japonicus is capable of attacking a number of other non-target hosts in the laboratory, and field studies now have demonstrated that there is some attack, and we can expect to see some attack under field situations. This chart shows the maze of potential interactions between 4 or 5 different species of native stink bugs and parasites and all the stink bugs that they can attack. So we can expect to see these kinds of interactions in the field. And this is something that we need to take into account in preparing our petition, which is about ready to go in. Going in the wrong direction here. Very recently, this nice study by Betsy Beer's group with a number of collaborators have demonstrated that in addition to successful parasitism, there's also significant impact on some non-targets from mortality due to unsuccessful parasitism. And this is another thing that we can expect to have to see in the field. But we also know from some of the studies that Sean Boyle did in his lab thesis work at University of Delaware and that Joe Kaiser talked about in his field studies, that there are behavioral screens that suggest that some of these non-targets will receive some level of protection due to the preference of Trissolcus japonicus for brown marmorated stink bug under field situations. So we're hoping that we'll be able to take all of this into account when we finally file our release petition and hope for the best. It's interesting to speculate on or to look a little bit at how other countries' regulatory approaches compare or differ. In Italy and Switzerland both, they've moved ahead with field releases of Trissolcus japonicus. In New Zealand, they don't have brown marmorated stink bug populations yet, but they've made the decision to authorize the release of Trissolcus japonicus in the event that brown marmorated stink bug shows up. In Australia, on the other hand, is much more restrictive. And so there's quite a range of regulatory views at looking at this. So we don't have time to discuss the further research needed, but I think that we'll get at this a little bit tomorrow in the discussion group. These are just some things to think about. And it's time for our questions. Thank you very much.
Thank you, Kim. While we're doing this, I'll just It's almost 5 o'clock. We're 1 minute before as far as I've got it here. We're scheduled for a 10-minute session, so I don't see any reason not to go a little past late or a little late here if people are up for it. With all those presentations, losing 10 minutes, that's not too bad. So.
Hey, Kim, this is Tom Kuhar. I can't actually put a question into the Q&A because I'm a presenter. So I'll just ask it if that's okay.
Sure.
Will the fact that some other countries have approved the release of TJ, you think that might influence the application here in the US?
Well, I think it's possible. You know, this is kind of a unique situation. The New Zealand decision to authorize, you know, a preemptive biocontrol is rather unique. I think our regulatory group in the permits branch has been willing to listen to new arguments. You know, they've already indicated that they're willing to consider allowing redistribution of populations from one state into adjoining states, which is a first for APHIS. So we'll put our best foot forward and make our best arguments, and we'll see what happens. You know, I'm cautiously optimistic, but you know, regulators do listen to people with adverse opinions, and it only takes a few people with very strong contrary opinions to shoot down a petition for field release. On the other hand, they also listen to stakeholders. And if stakeholders are active in making the case that, you know, we really need a solution and we need to have biocontrol as part of this solution, I think APHIS listens to that.
Nancy, if you could, if there's any questions that people have typed in, let's make sure we have those asked before we go on to the speakers asking questions.
Yeah, so a couple have just come in the last couple of minutes. From Mariana Zooks, I hope I said that right. Should we be focusing on pushing legislation that allows interstate movement of TJ instead of trying to release the ones from quarantine?
Well, in my discussions with Bob Pfannenstiel in the regulatory group, he thinks the best approach is to combine both in the petition for field release. And if they have an opportunity to authorize neighboring state releases without requiring a permit, you know, that could be done separately. You know, I'm not sure what their requirement is going to be for that. I don't know if anybody has approached them to ask, you know, specifically for release in a neighboring state yet.
From Max Ragozzino, when releasing TJ, what life stage is preferred, infested eggs or adults?
Well, you can make arguments for both. You know, in the long run, I don't know if it makes any difference, and probably it's best to use both approaches in different situations. You know, we have some evidence to suggest that when we release large numbers of adults, a lot of them get caught up in the wind or fly away, and they may not go where they're intended. You know, if we release them and the wind comes up, it may blow them in the wrong direction, you know, away from a refuge population. Some people have found that, or have proposed that if you allow them to emerge naturally from egg masses, you know, their behavior may be a little bit different and they might remain in the area longer. But if you do that, then they're more exposed to predators while they're sitting in their egg masses. So there are advantages and disadvantages to both approach. I don't think you can say that either one is not to be preferred.
And the last question from Don Weber. Kim, do you think there's a better strain in the Western US? Is there any comparative data that would allow direct comparisons based on the higher parasitism observed there?
Well, I think we don't have a lot of published data yet. Paul Abrams' lab is starting to look into this. You know, they're maintaining a number of different cultures. And this is an area that we need to continue to move ahead and do more research on. So I'm hoping that this is an area where, you know, we'll be able to get some collaborators together and come up with some of the answers to that question. I think that it may be that we'll have to provide some of that information for release petition if they ask us to come back and give more information. That's often an outcome when you submit a petition, is that they may be willing to consider, but they may want to see more data. So we've been through that with a number of different petitions. So we have to be prepared to look into that area.
Nancy, there was one more, I think, that it was from David Owens. Is it possible, or nope, that's the,
Yeah, that got answered by Ann.
okay, good.
That's my annoying cat in the
I
background.
have a question for you. And, looking at the results that Diane Alston had with the high level of parasitism on Catalpa compared to something else, how important do you think it is in terms of monitoring in specific trees for
well, it's definitely true that in any given area there are certain host plants that are more favored by BMSB for egg laying than others, and it makes sense to concentrate your surveys on those plants if your objective is to find the parasites, determine whether it's present or not, and its level of parasitism. But in the long run, there may be other reasons to compare parasitism levels in different plants or different habitats. I don't know if that answers your question.
Yeah, it just seems that it may be an important issue, the tree species you're monitoring in. And I'm just wondering how much of the variation among different locations could be due to that. And I was actually surprised to see that Tree of Heaven in Nicole's presentation. The Tree of Heaven was actually not that preferred compared to, what was it, black walnut, or I think.
Well, that doesn't surprise me. In Asia, for example, Tree of Heaven is really widespread, and it's not a plant that we go to when we want to collect brown marmorated stink bug or parasites on. I mean, they're much easier to find on other plants than on Tree of Heaven in Asia. So I've always been kind of surprised to see how much BMSB likes it here in the U.S. And I suspect that in any given location, it's the mix of plants, the mix of host plants in that area that determine which ones BMSB likes the most.
Any more questions for the speakers during the biocontrol session?
Jim, I had a comment, not a question. This is Larry. For Paula. So I think it's important to look at habitats and regional differences, but I think you have to be a little cautious. One of the conclusions you made was that the Great Lakes doesn't have certain parasitoids, anastasis and native Trisulcus, but if you have Mariana's data, that would be completely falsified because those were probably the most dominant ones in Michigan, which is a Great Lakes region. So you only had a couple Great Lakes states, and the difference were probably more related to the habitats those states sampled or something like that rather than a regional impact. So just a little cautionary note about conclusions that can be made.
Yeah, Larry, this is Paula. Yeah, I agree. And in the Great Lakes, I think there was only a riparian site that was sampled, and the numbers of egg masses was actually much lower than the other ones. And that's why I was kind of pointing out the kind of sampling effort, because I think that made a difference on what we might find in those different habitats. And it's changing over time. When you look at the data, I couldn't show 2017 and 2018. You can see even differences from year to year, but I think there was some really strong patterns, and then there was some, like you're saying, Larry, that we need to probably look at a little bit more.
Before we leave, I have one question, but it's for Kent Dane. Is that him underneath those bees?
Yes.
Oh, congratulations.
I like to mention that makes me ageless. That picture was taken 43 years ago.
Are there any questions remaining for anything else today?
Would you like to see the results from the prior biocontrol presentations?
Sure. Yeah.
I'll post. Let's see, Joe's.
Okay, we've gone through 10 minutes of discussion time here. And what's this one? I didn't see this.
That's the last one.
Okay.
Paula.
And so we're going to resume tomorrow at 1 p.m. Eastern Standard Time again. And we have one more report session on the management as well as economics and outreach component. And then we'll have the interactive session with the stakeholders. I appreciate everybody sticking around to the end. And I also, the behind the scenes efforts there by Kevin Judd and David. So if there's nothing else, we'll see you tomorrow.
Thank you, Jim. Thanks,
Thanks,
for all your
everyone.
help here.
Thanks, Jim.
Bye.
Thanks, Jim.